Hello, good morning, everyone. Good morning, everyone, and a very warm welcome. I am Dr. Namita Raitekar from School of Science, Sandip University, Nashik, India. It gives me an immense pleasure to welcome all of you for this international e-conference on emerging trends in bioscience and technology 2020 on behalf of School of Science, Sandip University, India and Biotechnology Society, Nepal. I welcome all the dignitaries, guest speakers from the national and international organization of repute, all the delegates coming from the different parts of the world for making this conference a global event. I also welcome all the panelists for this international conference. I think no lockdown can lock the spread of the knowledge, and education is highly infectious as it doesn't understand any boundaries and limits. That's the reason today people from all over the world are sharing this common platform to learn something new and gain the knowledge too. Once again, I welcome all of you and I would like to proceed with the conference. Now I will request Professor Vasudev Zamre sir, Dean of School of Science, Sandip University and convener of this international e-conference to address today's gathering. I welcome you, sir. Please proceed. Over to you.
Good morning, one and all distinguished guests, scholars, faculties, dear friends and fellows, ladies and gentlemen. It's a great pleasure and honor to welcome you warmly to the first stand of International In-Conference of the 2020 on emerging trends in bioscience and technology on behalf of School of Science of Sandip University, Nashik, and Biotechnology Society of Nepal. I take this opportunity to welcome our distinguished keynote speaker, Professor Liu Christopher from Biorefining World, United States of America. Additionally, I am happy to welcome Dr. Ramchandran Subramanian from Birla Institute of Technology and Science, UAE, Dr. Niranjan Koirala from University of Macau, China. Moreover, a cheerful welcome to Dr. Ram Bussell from Monash Biomedicine Discovery Institute of Monash University, Australia. I warmly welcome to Dr. Tista Joshi from Nepal Academy of Science and Technology, Nepal. To top of it, a friendly welcome to Dr. Raju Pratap Singh from Banaras Hindu University, Uttar Pradesh, India, and Dr. Gopalji Gopal from Uka Tarsadia University, Gujarat, India. As all of you are aware about the COVID-19 pandemic situation and facing its good and bad impact in day-to-day life, I think this conference will be a virtual platform for all students and scholars to interact with the eminent faculties and scientists from all over the world. The intentions of this conference is to connect all scientific community for addressing and discussing the recent issues, challenges in the healthcare sector, environment, and society. You are here today reflecting the importance of youth and their voices in this conference, as well as for absorbing the experiences, lessons from the eminent speakers. I am confident that during these 3 days, you will enhance and experience a different life, a different level of scientific zeal during the ETBT 2020. I'm very thankful to all the organizing committee and all the participants. I welcome you all again and thank you very much.
Thank you so much, sir. Thank you for addressing the gathering and explaining the motto behind this international e-conference. Now, this is a time to invite our keynote speaker of today's event, that is Professor Liu Christopher. Professor Liu Christopher is a founder and the director of biorefineries and working in Canada. He is highly accomplished and innovative professional with more than 20 years of academic and industrial experience. He is recognized by senior experts, peers, and employees for leadership, technical expertise, and operational savvy in development of nationally and internationally recognized programs in the science, technology, and engineering domain of renewable energy, environment, health, and sustainable manufacturing of bioproducts that support the transition to a bio-based economy and society. He analyzed complex issues and developed unique solutions which fully answer fundamental questions and advance current technologies. He has proven leadership abilities in managing complex multi-million dollar projects and cross-functional teams. He is aggressively promoting excellence in research and scholarship too. He has strong familiarity with federal funding regulations and the mechanisms. He is expert advisor in project and programs assessment for federal agencies including USDA, NSF, DOE, and EPA. He is a competent and prudent academic leader able to promote and foster a thriving research enterprise. He served as a director of two large multidisciplinary research and development centers. He is effectively leading teams in cutting-edge industrial research and conducting large-scale commercialization trials too. He has proven grant writing ability with a success rate of over 30% and funding in excess of $13.2 million. His exceptional R&D, publication, and innovation ability with more than 400 technical contributions made him an impeccable researcher and scientist. He has 11 patents to his credit, 4 books, more than 100 peer-reviewed papers, and more than 65 invited lectures delivered on several continents. I think this is just a brief introduction of Professor Liu Christopher, and after listening to him, we will come to know that how eminent personality he is. So I'm really happy and glad to have Professor Liu Christopher with us. I am inviting and I also welcome him for this conference. Now I invite him and request to deliver his talk on the circular bioeconomy in action, maximizing the value of renewable lignocellulosic biomass. I welcome you, sir. Over to you, sir.
Hello?
Sir, kindly unmute yourself.
Can you hear me now? Hello? Okay, so can I have the slides or should I share my screen from here?
Hello, sir, I will share your screen and you're audible too.
I'm talking about the presentation. Can you show the presentation or should I share my screen? Okay. Can you enlarge it a bit? Well, I hope you all can hear me and you can see me, and I would really like to thank the organizers for this kind invitation and the opportunity to share some of my research and recent work with you. And some of this work was done with my colleague Dr. Zambari as well, while he was still in my lab until last year. And I would like to personally thank him for organizing this event. And to be quite honest, I'm not very familiar with e-conferences, and but I hope I'll enjoy it because in this worrisome times of the pandemic, technologies like this enable us to stay in touch because research and education and student training should continue no matter what, and globally in each part of the world. Okay, so today I'm gonna talk about circular bioeconomy and the value of lignocellulosic biomass. And before I show you some of the research that my group has done in this area, I would like to give some definitions, especially for those of you who are not that familiar with this topic and terminology. Can we move to the next slide? Okay, circular bioeconomy. You can see it is the intercept between bioeconomy, which is, there are many definitions, but one of them is knowledge-based use of natural bioresources to produce economy goods and services in an environmentally friendly way. And circular economy, which is an economic system aimed at eliminating waste and the continual reuse of resources. So you can see that little diagram here that shows that products made then can be reused and remade, and that closes the loop. So basically, this is the concept. It's just that it is applied to bioresources, to natural and biological resources, and not to any other products. Not, for example, not to oil-based, fossil-based products, non-renewable products. So this is circular bioeconomy. So it's a very recent worldwide strategy to minimize and eventually replace the use of oil-derived products. And the focus is on sustainability of our biomass resources, and implies the reuse and the recycle of all wastes from biomanufacturing industries. So they're both economic and environmental benefits, and it creates high value from low-cost biomass wastes. It eliminates the waste, and it reduces the greenhouse gas emissions. These are some of the major benefits. And if you look at the slide on the right here, the bottom of this slide, this is another way to illustrate the circular bioeconomy. Bioproducts produced through manufacturing from biomass, and there is always bio-waste. And this bio-waste can be agricultural and food and agri-waste and municipal, and I'm going to talk a bit more about this later on. This bio-waste can serve as a raw material to produce new bioproducts back. So that closes the loop. And the technologies that are used, it's a suite of technologies used to convert biowaste to bioproducts, is called biorefining. Next slide, please. Okay, let's talk a bit about biorefining. So the transition to sustainable low-carbon bioeconomy requires the development of these bioprocesses, as I mentioned, through biorefining. Biorefining itself is defined as sustainable processing of biomass into a spectrum of bio-based products that can be a range of food, feed, chemical material, etc., and bioenergy, biofuels, power, heat, at a conversion facility that is called biorefinery. So, biorefinery, you can look here at the bottom of the slide, is analogous to the oil refinery, just starting with biomass instead of oil. And then the biomass is refined into the four major components of biomass, which are also shown here: cellulose, hemicellulose, lignin, and extractives. We are talking about production platforms of biorefinery. And then these platforms serve as feedstock for the for conversion of each component into fuels, chemicals, materials, and other value-added products, and heat and power. And the conversion platforms are chemical, biochemical, thermochemical, various integration, that's why the biorefinery is called also integrated biomass biorefinery, because all these platforms and conversion and production platforms are integrated in the best possible way to maximize the value out of biomass and wood and lignocellulosic fibers. Next slide. Okay, there are a number of advantages of biorefining. As I mentioned, they're all the products are renewable, non-toxic, biodegradable. The technologies are carbon neutral, and in some instances even carbon negative. And I will provide example for that. There is no effluent or any other type of solid waste generated. Everything is utilized. And all this enables the development of the circular bioeconomy, which is our topic today. Last but not least, there is no competition with the food supply chain, the so-called fuel versus food conflict, because the biorefinery utilizes non-edible, non-food-based biomass. Next slide. The biorefinery feedstocks, as we call them, or raw materials, you can see it's a variety of lignocellulosic biomass, starting with woody biomass, hardwood, softwood, and then these are just groups of, as they are grouped in different categories, energy and green crops. That will include grasses, switchgrass, miscanthus, and short rotation copies, agricultural waste. I just provided some that doesn't exhaust all the wastes, of course. Municipal and household waste, solid waste, sludge, food waste, etc., and industrial waste. Next slide. As you can see, most of the feedstock actually is waste. So that's why we're paying attention these days to biorefining and circular bioeconomy. So because of the time constraints, I won't be able to go into much detail and exhaust the topic. I'm gonna just provide some highlights on different in different research areas that this is our own research done. It's not based on literature. This is not a literature review presentation. It's based on my own research. Biowaste valorization, advanced biomaterials, biochemicals and natural bioactive compounds, and biofuels. So I'll go on each of these topics and give you some examples. Next slide. Thank you. So let's start with biowaste valorization. So, and let's take an example from the pulp and paper industry for waste streams that can serve as material for the circular bioeconomy. And the pulp and paper industry is an excellent example to illustrate this, because a number of processes that are involved in feedstock preparation and debarking and chipping and pre-hydrolysis and pulping and bleaching and paper making, they all generate some sort of byproducts or co-products or waste. These waste streams or side streams actually can be used to produce as a raw material to produce other value added products, which I listed here in general, and most of them are high value added products that are produced from very low cost waste products. So I'll give some examples with some of these with sludge and effluents and others. Next slide. Okay, this is a very recent work that we've done together with my colleague, Dr. Zambari, on zero waste biorefining of primary sludge. So primary sludge is basically waste stream of short fiber that cannot be retained on the paper machine in the pulp and paper making process, or cannot be retained on the washers and the wash screens, and it goes together with the effluent, and by gravitation is settled in the so-called primary clarifier, hence the word primary sludge. So it's basically lignocellulosic fiber that cannot be used for any pulp and paper product. It is a waste product for the pulp and paper industry, and the industry pays a lot to dispose of this material. Landfills or incineration, they're all very prohibitively expensive, and hence we did this research to see if we can make value out of primary sludge other than just burning the sludge or landfilling it. So a few steps of enzymatic hydrolysis to produce hydrolysates, then centrifugation to remove the solid, unhydrolyzed solids, and then the supernatant was subjected to ultrafiltration to separate the sugars as a permeate, and the sugars went to fermentation, and we produced bioethanol or biolipids using different microorganisms. Just to illustrate that this sugar stream is fermentable. We have not optimized it, but actually the yields are quite good, 92%, and the lipids are close to 40% weight-based. Then the retentate was actually recovered enzyme, and we recycled the enzyme. In fact, with the main reason being that enzymes are still expensive, and the more we can reuse the enzyme, the more viable the process becomes. So we were able to actually recover 40% of the initial enzyme load, which is quite a good achievement, 40%, which also makes the process much more viable. So we recycled the enzyme, and we also used the solids that were again a side product from the hydrolysate in experiments to produce bio-based reinforcement material that was compared to cement. And you can see the compression load was twice that of cement, so indicating that these solids can be used as a cement additive or reinforcement material on its own. So, having said that, you can see that from primary sludge, we produced sugars which were fermented, and we recovered the enzyme, and we also produced cement additive for reinforcement material. So there is no waste. This is a zero waste process. So the primary sludge is an excellent biorefinery feedstock because it doesn't require any pretreatment. It has very high carbohydrate content, low lignin content, and which is excellent feedstock for biorefinery applications. And obviously it will lead to a reduction of any production costs of biofuels or any other bioproducts, and it will be very environmentally friendly. Next slide. Okay, another example. This is a byproduct from pulp, from pulping again, from the pulp and paper making. It's called crude tall oil, which is recovered from the black liquor, from the pulping liquor, and can be fractionated into three major fractions: fatty acids, resin acids, and unsaponifiables. So from the fatty acids, we produced these products and the resin acids as well. We can see them better here: biodiesel, linoleic acid, fuel additives, and bio-based surfactants. So, like I mentioned, crude tall oil is a very low-cost product, five-fold lower than biodiesel and up to 150 times lower than fuel additives. So, again, the concept of valorizing, adding value to lignocellulosic biomass or derivatives of biomass. The price of linoleic acid is very high depending on the purity. That's why we also decided to purify linoleic acid from tall oil, and fuel additives, surfactants, again, are in demand, and the markets are expected to grow significantly. So here you can see how the bio-based surfactants were produced using fatty acids and taurine, which are all biodegradable, and producing a bio-based surfactant, which is also biodegradable. The linoleic acid required fractionation and the separation of the fatty acids from the rest, because it's a complex media of crude tall oil. So we separated the fatty acids, and then we separated the unsaturated fatty acids from the saturated fatty acids, and then through another process we extracted linoleic acid, and the yield was quite good, about 12% of the crude tall oil. In fact, that acid had a very high initial content, about 50% of the total fatty acid in crude tall oil was linoleic acid. So linoleic acid is also known as an essential fatty acid because it's not produced in the human body and needs to be taken as in the form of dietary supplement. It has many other health benefits as well. So once we extracted the fatty acids, the rest of the fatty acids were used as one example to produce biodiesel just by with methanol and using a lipase enzymatic catalyst. And fuel additives were also produced mainly from the resin acids, and we tested the properties of biodiesel, especially for countries like with cold climate countries like Canada and the Scandinavian countries. These properties are very important, and we've seen quite a significant improvement in poor cloud point, etc., C10 number, the ability to for biodiesel combustion. And of course, all these additives, as probably most of you know, they have beneficial impact on the performance of engines and reduction of emissions, etc. Next slide. Okay, lignin. Lignin is another byproduct that is becoming now commercialized from the black liquor again, pulping liquor, same as crude tall oil, commercialized in Europe and North America. I'm not sure about Asia and India, but there are a few plants that are making now commercially lignin, and of course there is a need for new uses and applications of lignin rather than just burning it. And until now, 90% of the total lignin is burned as a low-cost fuel. And the lignin is about 65 million tons produced globally per year. So this is huge. Only 2% are used for other purposes. So now research is being focused on use of lignin for a number of value-added products. There is a say you can make anything from lignin but money. So we are trying to change that. But there are problems and challenges with lignin because of the viscosity, the limited access to reactive sites, heterogeneous structure, polydispersity, high molecular weight, variation in quality of lignin and performance from one pulp and paper mill to another, because they use different wood species and feedstocks. So these are all challenges, and on top of that, difficult to characterize. Lignin has a very complex structure, and also some lignins, like the Kraft lignin, is not water soluble below pH of 10, so it has to be alkaline. This is scaling and hydrolysis lignin that is produced by enzymatic hydrolysis is not water soluble at all at any pH. So these are real challenges. Okay, so a number of projects that we've been involved, and I'm gonna just show one. Production of low molecular weight aromatics but we also have done work on seed additives from lignin, carbon fibers from lignin, and drop-in biofuels from lignin. Next slide. Okay, low molecular weight aromatics. So obviously lignin is composed of phenolics, the three types of phenol structures that actually can be obtained by degradation of lignin, and there are two ways of doing that: by biologically using lignolytic bacteria and fungi, or thermochemically using hydrothermal HTL is hydrothermal liquefaction and pyrolysis, and I've shown all this here. But the idea is actually to degrade it down to low molecular weight aromatics called BTEX. And this stands for benzene, toluene, and xylene, which currently are the main building blocks for a number of syntheses. And they currently are derived from oil. You can appreciate the idea that we want to produce bio-based phenolics, BTEX, and substitute that and replace the phenolic-based phenolics aromatics. So pyrolysis is one approach, which is thermochemical, that is actually performed at high temperatures and results in the so-called biochar, the solid phase, bio or liquid phase, and biogas, the gas phase. And these proportions between the three components can be manipulated, and depending on the conditions of the pyrolysis. And then the biool is the fraction of interest, because from biool one can extract some of the phenolics, which are degraded, you can see here. Or you can also see in the table about 26% of the bio oil consists of phenolics, low molecular weight lignin, which can be used for these applications, or also for many others, including replacement, let's say, of lignin, oil-based lignin in so-called phenol formaldehyde resins that are used to in plywood and oriented strain boards and others. The other approach, which is also thermochemical, is hydrothermal liquefaction. The advantage of this approach is that the presence of water is not a problem, because very high pressures are applied to produce the so-called biocrude, which is again a solution of low molecular weight lignin. With pyrolysis, the moisture content needs to be below 10%. Here it can be 20, 30 and higher, so there are no costs for drying of biomass involved in this case. Okay, the other approach is lignolytic enzymes as well, in addition to lignolytic microbes. These enzymes, mainly lacases, lignin degrading enzymes, lignin peroxidases, the so-called lacase mediator systems that are used to actually, lacase is oxidized by oxygen from air, and then lacase oxidizes a low molecular weight compound called mediator, and then the mediator penetrates the biomass to oxidize lignin, because where accessibility, barriers to access lignin in biomass. So this is the so-called LMS system. Of course, there are other enzymes that can also help assist in the degradation of the so-called lignin carbohydrate complexes in biomass in order to degrade lignin. Next slide. Another example, this time using effluents. This is spent sulfite liquor, again from sulfite pulping of wood, which is very rich in xylose, which is a pentose sugar. Xylose is an excellent inducer of xylanase and of another enzyme called xylose isomerase. So the xylanase is used, it has many applications, and you can see some the activities that were produced here on the sulfite spent liquor actually double that compared to a substrate like xylon. So it was very beneficial to use a low-cost effluent to produce xylanase. Xylanase can be used in the process called biobleaching, where xylanase is applied on pulp prior to bleaching, and that can result in reduction of bleaching chemicals. Some of them are chlorine, toxic chlorine-containing chemicals. Or it can improve fiber properties, or it can improve brightness, and many other applications. So, very important enzyme, together with cellulase for cellulose degradation. Xylose isomerase again can be produced on xylose, and then this enzyme actually catalyzes the conversion of fructose to glucose to fructose to produce the so-called high fructose syrups, which fructose is 30% sweeter than glucose. Also, xylose isomerase can actually convert xylose to xylulose, and then it enters the so-called pentose phosphate pathway to produce ethanol. That's another option. Next slide. Next slide, please. Hello? Okay, we're changing the topic. Advanced biomaterials from cellulose. I will provide 3 examples of textile fibers, environmentally friendly textile fibers, hydrogels, and bioplastics. Next slide. Okay, so these 3 markets are growing and drawing attention as potential biodegradable, renewable alternatives to the oil-based plastics, for example, or hydrogels. And the work that we've done here involves modification of cellulose in biomass, specifically in bleached pulps that have low lignin content. And I'm going to show you the basic reactions. Next slide. There are 2 main reactions. The first one is chemical modification of the fibers from bleached pulp. We can use enzymatic oxidation or periodate oxidation, chemical oxidation, to produce the so-called dialdehyde cellulose. Basically, the hydroxyl groups in cellulose are converted to aldehyde groups. The 2 adjacent aldehyde group, hydroxide groups are converted to aldehydes, and then the reaction is also shown here with pyridate and cellulose the aldehyde cellulose, and the byproduct is iodate, sodium iodate, from sodium pyridate, sodium iodate. So we can actually recover the pyridate by just using a low-cost chemical, which is available at pulp and paper mills, sodium hypochlorite, because the most expensive chemical in this is the pyridate. So a chemical recovery is a good idea, so we can reuse the chemical. Of course, the other option will be to use enzymes. Again, that will be a more expensive approach, but more environmentally friendly. So the second major reaction is cross-linking of the aldehyde cellulose with chitosan or another biodegradable polymer to produce another environmentally friendly cellulose that contains, it's a cross-linked cellulose, and the cross-linker is chitosan, and the cross-linking bond is this amino bond that is formed between the chitosan and the aldehyde cellulose. So, and only byproduct is water here. So this is in general, these processes are all patented, and this is as far as I can go in explaining the chemistry. Next slide. So the advantage is textile fibers. Why did we do this work? We did it because currently to make fibers, dissolving pulp is used and is converted to through a process called xanthation to viscose rayon, and this process involves a very toxic carbon disulfide chemical that is actually banned for use in certain in North America, in Europe. So this process that we've developed actually offers an alternative which is environmentally friendly, and it's an aqueous based non-toxic process, and it has other advantages like high fiber yields because we retain the hemicellulose, which is about 20%, which also results in cost advantages. And if you look at the properties like strength, and we compare our fibers to rayon, the rayon fibers, very similar. Of course, cotton is much higher in terms of strength, but cotton is, there are challenges with cotton and the production and the future use of arable land and all this. So our process can compete well with the rayon process, which is the environmentally unfriendly process we are targeting to replace. And another advantage is that the fibers can be spun on existing commercial spinnerets equipment, and of course this opens the door to introduce this process worldwide in countries where the carbon disulfide is banned. Hydrogels, well, baby pampers is a good example. What is important for hydrogels is that they have high water uptake, and currently they are produced from polyacrylic acid, which is again all-based compound. And You can see the water retention value, the ability to absorb water, is quite comparable to that of commercial superabsorbent polymers. And when it comes to saline water, which is the real condition for using of baby pampers and other hygiene products, bodily fluids will always contain 0.9% sodium chloride, so which is called saline water. So here actually our hydrogels outperformed any other, including the commercially available synthetic polymers. So again, the advantages will be that they're biodegradable and catalyst free and made from renewable materials and other advantages. Same with the bioplastics. Comparison here will tell you that the strength of our bioplastics was improved compared to commercially available synthetic plastics. So with the advantage, main advantage being that they're biodegradable. So the plastic bottles, for example, would take about 700 years to degrade compared to 7 weeks of bio-based plastic. So this is another hot topic for research and to clean up the environment from oil-based synthetic plastics. Next slide, please. Okay. I will just provide a couple of examples for production of biochemicals and bioactive products. Next slide. Lactic acid. We isolated a high-performing bacterial strain that can produce lactic acid at very high yields, high purity, and very high volumetric productivity. And you can see the lactate production here with this isolate Enterococcus. It's a sort of lactic acid bacteria, Enterococcus feculis, that we used, and we reached production titer of 180 grams per liter, which is quite significant in a fetch batch fermentation. But the most important probably result was the volumetric productivity of lactate that reached 10 grams per liter per hour, which is the highest ever reported, at least in the public domain, for production of lactic acid. On top of that, the purity was very high because the contaminants or so-called metabolites were less than 0.1%. And of course, if purity is high, then that will reduce the purification costs. In fact, the purification costs probably 70% of the total production cost of lactic acid. And lactic acid is used mainly for production of polylactic acid, a biodegradable polymer, and green solvents like ethyl lactate, which both have market growth of about 20% per year. So this is the polymerization scheme from lactic acid to polylactic acid, and we're developing the catalytic process that's gonna skip one of the steps in the polymerization and produce directly high purity lactide for polylactic acid. And the other advantage is that that can be done directly from the fermentation broth after the fermentation of lactic acid with this bacteria. So the purification costs will be significantly reduced using a catalytic reactor, and that's also patented. Next slide. Okay, bioactive natural products. We've done a lot of work, including with my colleague Dr. Zambari, on lichens and their pharmacological properties. We published also a few papers on that, investigating the antioxidant, anti-cancer, and antibacterial activities of these lichen species, and we published that as well. And you can see some significant results in terms of antioxidant activity, 89%, anti-cancer and antibacterial. Also, we studied the activities of corn cobs xylem, which is known as to have antioxidant activities, 86%, and antimicrobial half maximum inhibitory concentration, also quite good results. But in terms of anti-cancer research, this is the most recent one that we used a lichen species called Umbilicaria muhlenbergae, and through steps of extraction of lichens with solvents, fractionation, isolation, identification, analysis, chemical and biological, we were able to isolate one of the active compounds responsible for the anti-cancer activity of this lichen, at least on breast cancer, and 96% cell death is caused by this compound called gyrophoric acid, and this is a very promising result, of course, and this research needs to go to expand into studying other cancer cell lines, not just the breast cancer. Next slide. Biofuels, this is my last topic. I already mentioned about biofuels, and I'll briefly go through some of this research. Next slide. Okay. We've done extensive research to isolate new microbes that can be used for biofuels production. And for this purpose, we used extreme environments where you can find microbes that can survive these extreme conditions. One was the so-called Homestay gold mine in South Dakota in the States, which is about 8,000 feet, 3 kilometers deep, and very old gold mine. It's now closed, no longer in use, but it provides all these extreme temperatures and low oxygen and concentrations, etc. And the other one is the Yellowstone National Park in Wyoming, where we also did work with Dr. Zambari to isolate some extremophiles or thermophiles. Our interest was to find thermophiles that can produce thermophilic enzymes. So Yellowstone is one of the world's largest petrified forests, and with numerous hot springs and geysers, and it's a great location for isolation, to study the microbial diversity. And we used the classical isolation, screening isolation to isolate some of these thermophiles. Next slide. Okay, so to show you two examples, thermostable lipase, which was produced by Joe Bacillus thermodinitrificans AV5 is this one was isolated by Dr. Zambari, and I'm sure he remembers that, and we published that. And we use this lipase to produce biodiesel. And you can look at some of the, of course, it was identified 99% similarity with Joe Bacillus. It was identified as and pH optimum of 9. This is extremely high. I mean, alkaline, unique high pH optimum, retaining 90% activity at pH 10. Temperature optimum 65 and retains 70 at 80. This is a real thermophile, unique with thermostability at 50% at 80 degrees for 2 hours. The other one was, so this was isolated from Yellowstone National Park. This one is from the gold mine, the Homestake mine. And this is thermostable cellulase, also Joe Bacillus, just different species. And we use that for bioethanol production. You can see pH optimum quite broad, between 4 and 6, and temperature optimum 70 to 85, which is remarkable. And thermostability as well, very good at 50% at 70 degrees for 7 days. Next slide. So biodiesel, I mentioned already before that we produce biodiesel from crude tall oil, but here we use the lipase that Dr. Zambari isolated and characterized, and the lipase activity that we produced was on this is waste cooking oil, was 330 units per mil, which is one of the highest ever reported in literature for lipase, microbial lipase production. And of course, the actual biodiesel production using enzymes and in presence of methanol and triglycerides is quite well known, and the advantages of enzymatic biodiesel compared to chemical biodiesel are also well known in terms of biodegradability, non-toxicity, cost savings in terms of reusability and simplified process. If you look at the two processes, chemical biodiesel and enzymatic biodiesel, you can see that is much simpler and straightforward, and also with high purity biodiesel that does not require further wastewater treatment and additional costs. So biodiesel also, that's well known, has high energy density than ethanol, 60%, and 10% higher than biodiesel from vegetable oil if it's produced from crude tall oil. The greenhouse gas emissions of biodiesel are like 4.5 times lower than gasoline and 3 times lower than diesel, and 85%. So proven environmental benefits. Next slide. Biohydrogen. Also recent work. Hydrogen is the fuel of the future with the highest energy density known, and eco-friendliness, and it's mainly, unfortunately, currently produced chemically by methane reforming. 96% of the world production of about 45 million tons per year is by methane reforming, the other 4% by electrolysis, which is quite expensive process. So the biological and renewable alternatives here, and we biophotolysis, photofermentation, dark fermentation. We've chosen to work with dark fermentation because of the high yields and also the high substrate range that can be used to produce hydrogen. And these are two of the substrates that we used: municipal solid waste, remember, another waste that needs attention, and switchgrass, energy crop. Both have very high, fairly high, 65% biodegradable material, and of course present pollution burden, which is problematic to dispose of, and but on the other hand, they're very attractive as renewable feedstocks. So another isolate again from thermophilic isolate at 65 degrees. Here we biohydrogen production from switchgrass is shown, and we were able to reach the theoretical hydrogen yield of 4 moles of hydrogen per mole of glucose, which is great, and also about half that theoretical yield, about 50%, on switchgrass after on 5% switchgrass. So what is important here is that the switchgrass was not pre-treated even physically or chemically. It was directly used, and this hydrogen was produced directly from untreated switchgrass. So this direct bioconversion directly from untreated feedstock is called consolidated bioprocessing, or CBP. In one step, there are several processes involved, and what that the significance of that is that we can skip processes like pretreatment. Let's say if we use acid hydrolysis, where fermentation inhibitors involved. If we use saccharification through enzymatic hydrolysis, I mentioned is expensive, still enzymes are expensive. So there is a direct conversion of switchgrass or municipal solid waste to hydrogen. So skipping these two steps. So direct conversion, of course, that will improve significantly the cost of hydrogen production. Next slide. Bioethanol. I also mentioned that previously we worked with we invested a lot of work on bioethanol. Here, in this case, switchgrass, prairie crabgrasses, corn stover, these are agricultural wastes. They were used for pretreatment in this single screw extruder, which provides mechanical mixing and grinding of the biomass. Then there is an opportunity to add additional chemicals or enzymes during the pretreatment step. And if you look at the sugar composition before and after pretreatment with the extrusion, it's called extrusion, there is no change or loss of any sugars. There is no sugar degradation, and this is, of course, beneficial. And we used one of these isolates, Bacillus, to produce cellulases, and it was also shown that this Bacillus can produce ethanol as well in a single step, which again is called consolidated bioprocessing. Next slide. So, what are the advantages of CBP? If you look at this graph, the cost savings from CBP, using CBP, are between, in ethanol production, between 40 to 60%. If we eliminate the pretreatment, as I mentioned before, it's another 25% on average. If we use thermophilic consolidated bioprocessing. So CBP is here. You can see all steps can be done together. Pretreatment, enzyme production, hydrolysis, and fermentation, all 4 steps, compared to separate hydrolysis and fermentation that will require pretreatment, then enzyme production, then hydrolysis of these 2 major components, then fermentation, or simultaneous SSF, pretreatment, and then hydrolysis and enzyme production, and then hydrolysis and fermentation. So you can see how advantageous CBP can be. And of course, these are major advantages, and in terms of energy and cost and capital investment, it's called one-pot ethanol production. And of course, at elevated temperatures, the risk of contamination is much lower. And it has been estimated that CBP is about 4 times or more cheaper, less expensive than solid-state simultaneous SSFs, simultaneous purification and fermentation. Next slide. Okay, so this is, I'm almost finished. I'm wrapping up here. Of course, all these biorefinery developments, and this is all great, but there are still challenges which are related to market, the market, and the socio-economic and technical challenges for the biorefinery deployment. You can see here a drug can be discovered, and within 6 to 7, 8 years, it peaks and it pays off with a return on investment compared to a material or chemical, biomaterial, biochemical. It's going to take much slower, like up to 20 years for return on investment, for the revenue peak. Problems everywhere. Different industries don't actually, there is disconnect, agriculture, chemical, energy, there is some of the R&Ds of these industries are disappearing, and there is no dialogue between the customers and the producers. There is no premium for green products, at least in some countries. And in terms of techno-economical challenges, the conversion efficiency still needs to be increased and to reduce the enzyme costs, and integrate and reduce processing steps and energy demand and process streams and waste, as that was our topic today. And there are different systems of policies and regulations, and there are also concerns about deforestation, removal of biomass from the land, and pressure from environmental groups. Prices of these bioproducts vary up and down, and of course that debate that I mentioned, although it's not applicable to lignocellulosic biomass. And of course, if we look at the petrol, gasoline, oil prices, there is a like a threshold of about $50 to $60 per barrel, which below which the biorefinery is not competitive and will require additional effort and investment to become further optimized and integrated. Next slide. Thank you very much for your attention, and I'll be happy to answer some questions and to that was my slide, last slide. That was my group from my research group in South Dakota.
Thank you so much, sir. Thank you so much, sir, for your informative lecture. It was really interesting and informative session. I mean, it was amazing to know the various strategies and the technologies which have been involved in the processing of the solid waste, and we can convert the solid waste into the useful bioproduct. So it was really nice to know all the things related to this and the different concepts associated with it. So your tremendous work and the expertise in the biorefinery field is really of a great value to this conference and the society too. So we are really thankful from the bottom of our heart for being the part of this conference and sharing the content with us. So we are already running out of time, and but we have like a few questions have been raised by the audience. So we'll quickly have the few questions. So there is one question asked by the Dhruv Pandya, that is pyrolysis process is applicable to all the types of wet bio-waste. So I repeat, its pyrolysis process is applicable for all types of wet bio-waste.
Wet biomass.
Yes, sir.
Oh yes, yeah, it is. It is. The moisture content is not going to affect the hydrolysis process because the enzymes are so selective, and they go to the substrate. If we use cellulases, they will attack the cellulose component. In fact, there is a certain level of moisture content that is required for proper mixing and penetration of enzymes. So higher moisture content actually will facilitate the hydrolysis and the mixing, all this, yeah.
Yes, thank you, sir. So the next
Of course,
question.
if it's dry biomass, totally dry, that might be a challenge. So a certain level of moisture content is required. Even in solid state fermentation, there is certain level of moisture that is required. Yeah.
Yes, sir. Yes, sir. Thank you, sir. So the next question, which have been asked by Dr. Rishi Pagare, this is about the plastic problem is more, and what is your view on production of bio-based alternatives to petrochemical-based plastics, and are there any commercial technologies available?
I showed the slide that we worked on bioplastics, our group as well, and we have a patent on that. And there are many other groups working on this. As far as I know, it's not commercialized yet, but with all this intense research effort and focus on bioplastics, I'm sure, and there are huge companies involved in this research. Very soon we will have a commercial. In fact, there are some small, like demo scale trials at the moment, which will, of course, expand and go to commercial scale very soon.
Yes. Thank you, sir. So the next question has been asked by Dr. Naina. This is about most of the bio-based and or the waste-based technological processes are enzyme-based and becoming very costly due to the enzyme cost. So how to make these technologies as user-friendly and cost-effective? What is your view on it?
Well, I mentioned a few times that enzymes are still expensive. That's why we are applying strategies like enzyme recycling that will enzyme reuse so that we reduce the cost of the enzyme by reusing it multiple times. There are also other approaches that, of course, there are bio-based surfactants that also can be used together with enzymes. We did some work on that as well. And these surfactants, in fact, they act as lignin blockers. So they prevent the waste of absorption of enzymes to lignin, which is non-productive absorption, and it's a waste of enzyme. So by using these lignin blockers, we actually make the enzyme work on the substrate, and that's another way of saving cost in terms of enzyme. Of course, new enzymes which are more efficient and discovery of new thermostable, thermophilic enzymes, like I mentioned, that's another strategy that are more efficient. So bio-discovery and bio-prospecting of new enzymes is another approach. There are also non-enzymatic uses right now and developments using some, let's say, ionic liquids and which depultatic solvents that claim to be non-toxic and reusable, and these are also other alternatives to the enzymatic hydrolysis.
Yes, thank you so much, sir. Probably this will be the last question. This question have been asked by Professor Lab from USA. He is asking, can we produce biofuel from human stool?
Can you repeat the question?
Yes, sir. He is asking, can we produce biofuel from human stool?
Biofuel?
Yeah, biofuel from human stool.
Yes, we can. Yeah, of course. It's a waste, organic biological waste, and the easiest way is to produce biogas, which is biomethane by just anaerobic digestion. I think in India that's quite popular.
Yes, sir. Yes, sir, definitely.
Domestic. Yeah.
Yes. Thank you so much, sir. With this, I think we can conclude this question answer session. And thank you, sir, for being with us and for this such a wonderful lecture. Thank you so much.
It was pleasure to be able to share my work and to contribute to your conference. And I wish you best of luck for the rest of the conference. I'll try to join and to participate, but there is quite a time difference. Here is about 1 o'clock after midnight.
Yes, sir. Yes, sir. We can understand. Thank you so much, sir, for taking this extra efforts for us and just awaking at this midnight and working for us. Thank you so much. We really appreciate.
Pleasure.
Really.
Pleasure. Thank you.
Thank you. Thank you so much, sir.
Bye-bye.
Bye-bye. Hello, everyone. Now this is the time to introduce our next speaker of the today's session, that is Dr. Ramachandran Subramaniam. I invite Dr. Ramachandran Subramaniam. He is Associate Professor in Department of Biotechnology, Birla Institute of Technology and Science, Dubai, UAE. Sir has completed his ME and PhD in biotechnology. He has also completed his MBA in human resource management. Sir has more than 20 years of experience in the academics. He is really eminent academician. He has taught to the many courses as at postgraduate and undergraduate level. His main thrust area of research are microalgae, biodiversity and mass cultivation of microalgae, plant biodiversity and conservation in arid environment. He is also interested in biomass-based biofuel production technology. Sir, till now, has supervised more than 70 research projects at undergraduation level, post-graduation level, and as well as he is having PhD scholar under his able guidance. His main area of the work is environmental biotechnology, molecular biology, and proteomics. He has guided several dissertations and the thesis on these topics. He also has few more research projects in his credits. He has many funded research projects with him. The sir has extensive contribution in the research and he has many research collaboration at the national as well as international level. He has been awarded by number of award and prizes. Sir has been awarded as a Fellow of Academy of Science, Animal Welfare, Society of Toxicology, India in 2008. He has been also awarded as a Young Scientist Award in 2008 by Federation of European Microbiology Society, UK. He has also awarded as a Senior Research Fellow by the CSIR Council Government of India. Before that, he has worked as a research fellow and the junior research fellowship from BITS Pilani. He has received many best poster presentation at national and international conferences. He is also working as a professional member in many professional bodies. He has been invited as a guest speaker and resource person at national and international level. He has been visited to Italy, USA, and Australia for his work and his excellency in his work. He is a part of many academic committees and as well as serve in different schemes and services. He is a part of different meetings and the academic participation too, especially in the engineering students renewable energy competition, UAE University. He has arranged, organized number of conferences and symposia as well as workshop in his tenure. He has a great contribution toward the research and he has one patent to his credit and 3 book chapter. Also, he has more than 20 publication in his credit, which has been published in the International Index Journal with a high impact factor. Not only that, Sir has published 3 laboratory manuals on the genetic engineering, industrial microbiology, and downstream processing. The important thing, till now, he has submitted 24 gene sequences to the NCBI site, and he has not only worked for the curricular activities of the student, he has also worked tremendously for the extracurricular and co-curricular activities of the student. Till now, he has participated in 39 international and national conferences and he has guided number of teams, a students team and for the competition and most of them have award winning. So this shows that he is a very dedicated and passionate teacher and the professor and know how to shape up the career of the student. So with this brief introduction, I welcome Professor Ramachandran Subramaniam sir to initiate his talk on Penicillium purpureum, which is a Napier grass, his varieties for the animal fodder production. I welcome you, sir.
Okay, very good morning. I hope I am audible. Hello, am I audible now? Am I audible now?
Yes, sir, you're audible. I have
Okay.
shared your screen and you can
Yes, yes.
continue with the presentation. Thank you.
I'm waiting for your presentation, please. Okay. Okay. In the meantime, very good morning, all of you. I think this is a very challenging time, and organizing such a conference in a very short notice and with a very good participation around the world, it makes me also really very happy to participate and contribute. I think one part of the work what we are doing here in UAE. I think yes. Okay. Okay. So to start with, I hope we are all focusing mostly with respect to growing population, poverty, hunger, how to feed the hungry in the next 20 years or next 50 years or possibly next 100 years, like that. We have some projections from like World Health Organizations, United Nations, like that. We are talking much about that, and especially during this challenging time, like for example in this COVID, I think entire transportation is completely stalled. Now all are being fed with the available resources. So now in case unless we have sustainable take example food production, especially with respect to local food production I'm talking about, we may not be able to feed even the human, and that's what it is. With rapid industrialization, I hope these days they are focusing intensive agriculture production systems. Intensive agriculture mostly focusing towards indoor agriculture production. Here in UAE, I have a really very good experience how they make use of how they make use of indoor agriculture production to feed the local population. Uh, I hope there is some Photoshop, something is appearing there in the. Okay, okay. So you are taking care of. So here it is being mostly arid environment. They focus mostly with respect to indoor agriculture production system. For example, here you can see what you see here in the stores. Let it be cucumber, tomato, lettuce, leafy vegetables are all being produced locally with the indoor agriculture system, like a greenhouse. We have very highly advanced agriculture, irrigation systems, so that we can produce such kind of agriculture produce using indoor environments. But we are giving much emphasis with respect to feeding the humans. But parallelly, we need to work on with the livestock population as well. I hope in India or in the around the world we can see nowadays mostly concentrated animal livestock production. Let it be chicken, let it be dairy farm is for milk production or meat production. It is being commercialized. It means in a very small area. Thousands of take example cattle or sheep or chicken are being confined, and mostly they import animal feed so that to feed the animals. But when you see here in UAE, take the example in the case of 2016 data. As per the 2016 data, roughly 5 million livestock population, that includes cattle, camel, sheep, and so on. As per the 2015 data, more than 200,000 tons of animal feed being imported. It means now you see here with respect to, because here local production is very very less, so it means we depend mostly animal feed produced, take for example in Africa, European countries, or even mostly from India, where here in several industries, especially dairy farms, are now importing such kind of animal feed, mostly in the processed animal feed, for example, as silage we call it. So now here to encourage local production, here fodder crops, for example, Rhodes grass, alfalfa being encouraged, but these are all very water intensive crops. So to minimize water use, the local research organizations are giving much emphasis to use the saline water. I hope here you all may be knowing the soil also saline, and also the water is highly saline. So to how you can minimize the freshwater usage, because here we produce freshwater using desalination plants. So then we meant for all domestic use, let it be for human or industrial use, and also for agricultural use. So we cannot afford freshwater for such kind of large scale agricultural production systems. So what they do is they mix with the fresh water with saline water, so that they test a number of crops using a blend of, it means mild to moderate saline waters for agricultural production system, especially in the open and rural agriculture system. Here you can see such kind of highly salinity tolerant crops. For example, it may be quinoa. I hope many of you know that quinoa, barley, salicornia, like this. You have number of salinity tolerant crops being tested here for use for both human applications, for food applications, as well as for forage applications. We focus with respect to fodder applications. We have collaboration with local research organizations, and we tested a number of forage crops, because we may not be able to focus different, but I focus with respect to one area, that is fodder crops, and that too that can have multiple uses, not only for fodder applications, possibly for bioenergy or textile applications. I can tell you that it means my talk is a little bit simplified, because Professor Liu Christopher outlined various uses of biomass for other applications. It may be bioplastics, biofuel, biohydrogen production, textile applications like that, and so on. We outlined. So I don't like to give much emphasis with respect to the other uses of biomass as such. But next slide, please. My topic is with respect to the Napier grass for fodder application, and also this Napier grass, they call it around the world differently. Like for example, giant king grass, elephant grass, or Napier grass. Sometimes Pennisetum purpureum as such. So, but this is a multi-purpose grass, and it's very high yielding. I hope several of you may be knowing who is staying in India, because this is one of the cut and carry grass, they call it. Mostly the small farmers and the agriculture universities encourage the small farmers to grow this grass in their backyard, so that they can cut and feed the one or two cow what they grow in each house. Okay, so here in UAE, to reduce the dependence of import of animal feed, now the government is encouraging, and I hope now many of you may be knowing food technology challenge. There are millions of dollars worth challenge. Possibly they have postponed the event as such, but otherwise we also submitted proposals on that front as well, because they encourage a lot of local production for sustainability. Take for example, in the case of COVID, it means they are realizing, it means local production is very, very important to meet the growing demands for the human as well as for animal as such. Okay, so now when you see here biomass, it means a number of fodder grass species being tested here in UAE. Like as I mentioned, like alfalfa, Rhodes grass, centuries species like that. But I brought some of the fodder grass like multicut fodder sorghum, centuries, what's called fodder sorghum, guinea grass, napier grass, like that. I brought some good number of fodder grass species. We tested, and out of so many different grass species, we tested multicut fodder sorghum, guinea grass, and napier grass found to be really suitable in this environment. Here we are growing this napier grass for more than 7 years in the controlled environment. Like controlled means we didn't release to any farmers. We are testing. Now these grasses very well adapted to this environment. Here I hope many of you know that temperatures mostly during April to October, it peaks up to 48 degrees centigrades, and during winter seasons it may go to roughly 2 degrees, like that. This environmentally, it means this temperature range fluctuations, and it's really arid environment. That's what I can mention you. So as I told you, that it means we tested a number of grass species. We narrowed down to napier grass because it can produce very large amount of biomass under well-fed fertilized conditions. It can go around, it can yield around 400 dry tons per hectare per annum. This is what is published data shows. But as per our results, we could reach around 250 dry tons in this environment. That's what we could achieve here because of this water shortage, temperature, environmental impact. Here soil is mostly sandy soil, not like very, very rich soil, as I can mention. So now this biomass can be used as a kind of fodder, bioenergy application, lignocellulose, what Professor Liu mentioned, for lignocellulose can be used for bioethanol production, and so on. Okay, can also be used for natural fibers, for papers and textile industries. As the next slide, please. Okay, so now when you see here, selecting the crops, as I mentioned to you, that it means it should have low fertility requirements, salinity tolerant, and low energy cost for planting and harvesting, especially towards biomass for multi-purpose applications. It may be bioenergy or fodder applications like that. Here we found that, as I mentioned to you, that it means napier grass, it can grow in because it's well adapted to this environment. And we tested with respect to growth characteristics, nutritional properties, fiber content, energy content, approximate and ultimate analysis of the biomass. Next slide, please. So this napier grass here in this, you can see the photograph is grown here in the International Center for Bioscience and Agriculture. We have dedicated plot for testing and growing continuously using saline water as well as fresh water. Here in India, it can grow maximum height of 15 feet, but here it is growing 8 to 12 feet high. It's growing because subsurface there is no fresh water. We use only drip irrigation. So beneath 2 feet depth, you may not see any moisture content, but still this grass is having a wonderful root system. It can go and reach up to 2 meters depth. I think you can cut the grass repeatedly every 45 days. You may be able to get fresh fodder, and in a year, it means you may be able to get roughly 4 to 6 cut you can see, but up to 7 cutting also we can reach in case if you are cutting very every 45 days like that, in case if you are under optimum conditions. And when you cut it means it quickly regenerate. I can show you some of the photographs in the subsequent slides as well. It has very succulent stems and lush green. When you feed animals, it eats everything without any wastage. Next slide, please. So this is the one first we after the planting, you can see how nicely is growing this napier grass. When you see the nutritional qualities of this grass, The crude protein is roughly 11% to 14%. It varies. We grow these two grass varieties, Co-CN4 and Co-BN5. These are obtained from Tamil Nadu Agriculture Research Institute. We have collaboration with the Department of Forage Crops, and it has very good fiber content. We use this also along with other crops, for example desmanthus, cereals and pulses, and to make animal feed. So we are not having what's called immediately feeding animals. We process into dry pellets and we store it. It means we may be able to feed when we need, as and when we need, we may be able to feed the animals. Next slide, please. We tested the calorific value and ultimate analysis of the raw biomass for because we grow in salinity as well as freshwater grown, and there is not much significant variations between the salinity grown and freshwater grown, because we didn't grow this grass in the high salinity, roughly greater than 14 decimen, because it can grow only up to 7 decimen without much impact, but above 7 decimen means it significantly affected the growth of the grass as such. So we grow maximum of 7 decimens, but for animal fodder applications we never use saline water. For fodder application we use only the freshwater for biomass production. But whatever the case, we didn't see much significant differences in terms of CHNS analysis, and especially there is no sulfur found. So it means in case if you are using it as a kind of biofuel, as a pellet, it is not going to make much impact with respect to the coal alternative. That's what it is. Next slide, please. Next slide. So the thermogravimetric analysis of this grass didn't show much differences as you seen in the previous slide. Because it's possibly exclude or extrude the salt in the soil itself. So only possibly it's taking up only the water. There will be some kind of mechanism. We are not studying the mechanism of how this salt tolerance is happening there in Napier grass, but we are working with respect to biomass applications only. So next slide, please. The elemental analysis of the biomass, we tested mostly in case if you are growing any grass using saline water, we may be in position to test the sodium and potassium content. So again, this data shows that not much significant variations in terms of sodium content. So this also implies that it means all are within the permissible limits for use as a fodder as well as biofuel applications. Next slide, please. This is what the layout of the Napier grass field studies which we carry out. As I mentioned, it means we continue to replant and then we test, replant and then we test, so that the grass is getting established over here, and there's not much significant variation from year to year after the first 3 years of planting. So now it is, as of every year, if you're planting, there is not much impact with respect to growth of the grass as such. This is a randomized custom block design for testing the salinity tolerance of the Napier grass varieties. Next slide, please. Okay, so you can see here every year or wherever we test the grass, how we prepare the land area, irrigation systems, and the planting and growth of the Napier grass, it quickly establishes. You see here within 1.5 months it grows very fast, and initially maybe it will taking some 2 to 3 weeks to show the shoot, but after 4 weeks it rapidly grows in this, and because the soil is very sandy, the root system established very fast. Next slide. As I mentioned to you, because the Napier grass is mostly fiber-rich grass species, and we blend with the desmanthus. This desmanthus is a desmanthus variegatus species. It's a highly protein-rich fodder. It has roughly 14% dry weight. in terms of total protein content. So we mix the nappy grass with the Desmanthus species, and it's a leguminous plant. It once established, it grows for 30 years. So we don't like to replant again, and it very nicely it established. I can show you the next photograph. How it lush growth of Desmanthus here. So we parallelly grow nappy grass and nappy Desmanthus, so that we can test the use of nappy grass blended with Desmanthus for animal feed application. Next slide, please. So the biomass has multiple application, as Professor Liu outlined. It means the biomass once fully grown, not in case if you are using as a kind of biomass, it can be used tender 45 days to cut it. But in case if you are using it for any other application, it may be a biofuel or fiber or lignocellulose, to fully allow the grass to mature. So 8 to 10 months it takes. So it means 8 to 10 months mature grass being used for extracting the cellulose. You can see how beautifully you can get the cellulose. It's like when you see here woody wood derived lignocellulose, we can get roughly 40%. We can get in the case of nappy grass, we can get up to 40%. It's comparable in terms of woody biomass derived in terms of quality and consistency of cellulose derived from this nappy grass. As the next slide, please. So this is the, if I'm not wrong, the last slide also. Nappy grass can be used for developing biochar, bio oil. We produce bio oil. Is already communicated this paper also with respect to CV, GCMS analysis, FTER, complete analysis of the bio oil derived, and cellulose, as just you see in the previous slide. Animal feed in combination with other grass species and leguminous plants can be used. It's a multi-purpose, as I mentioned to you, and also large biomass can be harvested. Next slide, please. So major outcomes, when in case if you are harvesting freshly, it can yield 61 tons of green fodder in the first 5 months. Overall, in case we are harvesting, it reaches around 250 tons. Now we are focusing with respect to nutritional balance animal feed pellet production for local applications. Next slide, please. So we thank Dr. Prakash Kumar, Department of Chemical Engineering, Dr. Neer Sood, our collaborators, Dr. Lyra and Dr. Khalil Amar of ICBA, and also our collaborators in Bich Pilani, Hyderabad campus, as well as in Pilani campus, IIT Bombay, and Dr. Bhamendra, and a number of research scholars and thesis students worked on this project. And we thank all people involved in this project. Thank you so much. Any questions, please? Any questions?
Yes, thank you so much, sir. Thank you for your informative lecture. It was really wonderful to know how this Napier grass and their varieties can be used for the different applications, and the biomass can be processed. Thank you so much, sir.
Thank you
So
so
there,
much.
so there are few questions which have been raised by the audience. I will just take the few questions. So there is one question about, does the salinity of soil affects or alter the nutritional content of this Napier grass?
To answer this question, normally for preparing the soil, we flood the soil with the fresh water, so that is as already it's a sandy soil, so it just like it seep out, so it takes out all the salinity down. Then we plant. So in case if you see the, that's why, as I mentioned to you, for fodder applications, we never use saline water. Though it is not accumulating salt on its biomass as such. But for animal fodder application, we don't use salinity at all. For biofuel applications, yes, we have tested up to 7 decimals There is not a significant impact with respect to biomass yield altogether. And also the elemental composition, CV analysis, thermogravimetric analysis, and also not much impact. That's what we could observe.
Okay. Yes. Thank you, sir. Thank you so much. So the next question is, does it require any special supplements for the proper growth and to get the maximum productivity?
So far, here what we have grown is only manure. It means farmyard manure, we call it. We get the farmyard manure from the local dairy farms. It is not. We didn't use any fertilizer at all. Only soil preparations added with manure. So that with that input, we could achieve 250, approximately 250 dry tons per hectare per annum. But in case we are using any fertilizers, possibly we can see some increase in biomass. But there is something I want to mention you here, because the temperature here during summer is very high. So the use of fertilizers that may give you like a very tender biomass, that may in turn heat injuries we may observe. That's what the trials what we tested. The fertilizers used is affect the overall biomass during summer seasons. That's what we didn't use fertilizer at all. We use only the manure so far. That's what I can tell you.
Yes, sir. It means, does it economical?
Yes, it is 250 tons is really huge. When you see here, centuries or any other grass, what we tested here, even the International Center for Bioscience Agriculture also very much keen, because all other biomass, let it be centuries. I am giving you one example. It's giving 3 to 5 dry tons per hectare per annum. But here is something is 10 times to you got no. I think it's more than 100 times. I can say that in terms of overall biomass is a concern. Total overall yield I am talking about.
Oh, very nice. Sir, the next question is, can you just highlight any product other than the fodder which is commercialized by using the Napier grass?
So far, a number of, even in IITs, Pune, IIT Chennai and all, they are working very rigorous on the use of Napier grass for bioethanol production. But so far, it's not commercialized. But in Europe and in Thailand, they are using giant king grass for biogas production, and also direct combustion of the biomass for energy production, like co-firing, we call it. But other than these biofuel applications, so far lignocellulose derived from napier grass for bioethanol production is not very much successful, because I hope even woody biomass also is not very much successful, not commercialized. But in European regions, you can see giant king grass very popular, very popular. They use this giant king grass. It's nothing but pure napier grass only. So for biogas production, and they say community level energy production. Community level means something around 100 to 200 homes. They have some dedicated land area for production of napier grass. They collect the waste from the community level. They mix with the napier grass that act as a kind of base and the microwave inoculum, and that converts the napier grass for biogas production as well. I think you can get a number of literature and commercialized systems. Commercialized system, it means production systems for biogas production. That's what I think. Possibly this is a good time. Even in India, they can make use of this wastewater for biomass production, and then they can make use of biogas for feeding, producing electricity as well.
Yes, thank you so much, sir. So we'll just quickly take the last question. This question is from Chetnath Naupane from Nepal, I suppose. He is interested to know what is the possibility of the grass to grow in Nepal.
It's highly possible. That's why here, Thailand, Europe, here is highly what's called arid environment. In case, if I think some of the Kathmandu University faculty members, they came here, visited the fields also. I was really very happy. They were briefing. But anywhere it can grow. This is a grass. That's what I'm telling you. This is a grass. Anywhere it can get adapted. That's why here in the area environment it's getting adapted, and that too in Nepal and also possibly is a very good climate. Possibly it will very nicely it will get adapted. That's what it is. In case they want that fund, I may be really very happy to help them also, please.
Yeah, thank you. Thank you so much, sir. Thank you so much. This was really informative and insightful lecture. We are really delighted to listen your talk. I mean, this the way you highlighted the importance and the this new sustainable approach. It can be any condition, economical crisis, or anything can be suddenly come out. But I think we must be having such sustainable approaches, and we can be prepared to fight with any of the crisis with such kind of approaches, that for sure. So thank you so much, sir, for highlighting the importance of this sustainable approach and the commercialization point of view as well. So thank you so much, sir, for being with us and joining us for this today's event. We really appreciate your presence for this international conference. Thank you so much.
Thank you so much, please.
Yes, bye bye, sir. Take
Thank you.
care. Yes, with this, I would like to inform that the speakers and their keynote speaker and the invited speaker, their lecture session has been completed, and very soon we will be starting with the presentation session. So all the participants who are going to present their poster presentation or the oral presentation, they should be ready with their presentation. We can have the 5 minutes break, and after that, very soon, we will be starting with our presentation session. Yes. Good afternoon, everyone. We'll initiate with our presentation session. As per the instruction given to all the participants, please stick to the timeline. Yes, the first presenter we have is Jai B. Pandya. Yes, Mr. Jai B. Pandya. Yeah, you can start with your presentation. I will share your presentation.
Hello, hello.
Yes, hello, sir. You can start with your presentation.
Hello.
Yes, hello.
May I audible?
Yes, you are audible. You can start with your presentation. Let me tell you, there are a panel of judges and they are going to evaluate all the presentation for today's session on the basis of the content which is there in the presentation and your skill, presentation skill, as well as the understanding of your topic. Okay, so all the best.
Yeah, sure. Thank you. Hello. Good morning, all of you. My topic is abiotic factor of analysis of seawater and coastal soil from different area of Bhavnagar district, Gujarat, India. Why I have choose this topic? The main reason in Bhavnagar area there are uh, 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 so many plants, but the marine plants are very less. We have located Khoga, Kuda, Kodiak, Hathap, Mitividi, Gopnath, and Janjmer. Here we want to grow some. Hello, yeah. Here we want to grow some kind of mangroves as well as the marine algae. So for the development of the farming and for the protection of coastal area, we can start a development of mangroves. So in the water analysis, we have checked the pH, temperature, electricity, salinity, total suspended solid, acidity, alkalinity, chlorinity, total hardness, calcium, magnesium, ions, and dissolved oxygen, biological oxygen demand, chemical oxygen demand, and in the soil analysis, we have performed pH, temperature, EC, salinity, chloride, calcium, magnesium, soil moisture, soil porosity, soil holding capacity, soil organic carbon, soil buffering capacity, as well as the soil texture that is porous, gravel, fine sand, slate, and clay. What is the problem of the Bhavnagar coast? If we see the other area of the Gujarat coastal coast, uh, it is very easy to grow the marine algae as well as the that mangrove cultivation, but in Bhavnagar coast, very less amount of the mangroves and marine algae are present. Only in the Gopnath region, that some mangroves and marine algae are present, but in other areas such as Ghoga, Kuda, Kodia, Khathab, Mithivedi, and Janjmer, the uh, uh, marine algae as well as the mangroves are absent. So we are trying. No efforts have been made by the local people to grow the mangroves and marine algae. So we have analysis that if we see the parameters for the seawater, the pH is almost equal in all the locations, 7 locations, that is Ghoga, Kuda, Kodia, Khathab, Mitivirdi, Janjmer, and Gopnath. And temperature is not having much more variation. But electricity, EC, electrical conductivity of that locations are quite different. That is not big change, but when we see the TSS content, total suspended solid, in Kodiak and Hathab, the TSS content is a lowest most, while in the Janjmer and Gopnath, it is near about it is about 190 and 195 mg per liter. So in that case, if we see, there is a chance to grow the mangroves and marine algae in that region. We can say the acidity is higher in the Kodiak and Hathab region. Uh, in other location, it is normal, while alkalinity is lower at Hathab region. The chloride content is about 15% in Kuda, Mithividi. Hello, hello. If we see the hardness level is 100 mg per ml at Mithividi, while in other cases it is above the that content. The calcium level is less at the Kodiak, while magnesium is higher at the Goga and Kuda, while in Kodiak, Hathab, and Mithividi it is in very less amount. So if we see the DO, BOD, and COD, it is very less in CUDA, but in BOD it is higher at Korea, Khathab, and Mithivedi. If we see the coastal soil parameter, the pH is higher at Janjmer and Gopnath. It is around above 8. The temperature is near about 25, ranging between 21 to 26 degrees Celsius. The EC of that area, the EC is higher at Mithividi, Janjmer, and Gopnath. The salinity of the soil is near about equal, while the chloride content, it is uh, maximum at the Kodiak region. The calcium ion is higher at the uh, Janjmer and Gopnath, while magnesium ion, it is high at the Hathab, Mithividi, Janjmer, and Gopnath, while very less in the uh, Goga, Kuda, and Kodiak. The soil moisture content, it's very less at the Janjmer and Gopnath. The soil porosity capacity is around 12% and 15% at sequentially Chanjmer and Gopnath, while soil holding capacity is maximum, also maximum at around 25 and 27 percent, while organic carbon, that is highest at Ghoga, and soil buffering capacity of NaOH and HCl, if we observe, it is near about equal in all the regions. If we see the now the parameter of soil texture, if we see the soil texture, the porous, gravel, fine, and sleet and clay, these all are higher at the Janjmer and Gopnath. So in the view of this observation, if we see the Gopnath, it already have the mangrove patches, but Janjmer, Mithividi, Hathap, Kodia, Kuda, and Koga, it doesn't have the mangroves and marine algae. So, if we want to cultivate the Goga, Kuda, cultivate the plants and algae at these other different areas, the Kodiak, Hathab, and Mithividi, these are also the suitable area where we can grow the mangroves and cultivate the marine algae. Even though uh, we can plan the uh, uh, farming of the marine algae at this region. The Kappa ficus we are mainly planting in the Halophilic area, and hello, hello, am I audible? Hello,
Hello. Hello. Yes, right. Your time is finished. Have you completed with your presentation?
H- hello.
Yes, sir. Can you hear?
Yeah, now it is audible. We are having a heavy rain, so it is disturbance in the network.
Okay, yeah.
Hello.
Yes, your voice was not there for some time. So have you
Yeah,
finished with
definitely
your
because
presentation? Yes.
yeah, I have finished. Yes.
Yeah, thank you.
Okay.
Yes, the session is open for discussion. If anybody is having any question, please drop the question in the chat box. We can wait for 1 minute. If there are any question in the chat box, then we'll have them. Audience are requested to post the question in the chat box.
Hello. Hello.
Yes, sir, you're audible.
Yes, it is also a request. Hello.
Yes, sir.
It is a
Yes, sir.
request. If any suggestion is there, it is advisable or very valuable for us to go further for the cultivation purpose. Thank you so much.
Yes.
I think there was a interruption in between, so there may be no questions.
Yes, sir. There is one question. Can you please
Okay.
highlight other applications? There is a
The
question from
application of this, yeah, the application is that we can nowadays the cultivation of marine algae, it is blooming in Gujarat as well as the uh, Tamil Nadu and Kerala, but in smaller patches, not for the entire region. Gujarat having a 1600 kilometer area of coastal area, but out of that only 50, 60 or 100 kilometer is cultivated for the marine algae. If we try at the different region, what are the possibilities or what will be the outcome, we can plan according to that. And this is the initial work we have done. What are the preferable area at our location we can grow the marine algae, as well as we can improve the cover of mangroves, so from the natural calamities we can protect the Gujarat. For that purpose we have done this smaller research work. Thank you.
Yes, sir. There is one question from the panelist side. Uh,
Yeah.
yes, the question is can you just explain how the habitat or the plant diversity can be affected by this quality of the seawater? What can be the probable mechanism behind it?
Thank you, panelist. Not only for the parameter of seawater, but we have to combine with the soil structure and soil texture. With the salinity, we can, if we plan for the cultivation of
Good morning. So we are done with the first presentation from Pandya, Mr. Pandya. So I would
hello.
now like to request the next participant, Dr. Sandhya. Hello, Dr. Sandhya, you are on board with us in the studio. I will be sharing a screen with you. Please,
Okay, sir. Okay, sir.
uh, once the presentation is visible to you, kindly proceed with your presentation, and you have the time of 6 minutes.
Okay,
Kindly
sir.
take note of your presentation time. So we have with our studio our Dr. Ravi Kiran Pagare as a panelist. Welcome, Dr. Ravi Kiran Pagare. You are on board with us. Dr. Sandhya.
Hello.
Yes, ma'am. Yes, just please wait 10 seconds. We are just redirecting you to the
Okay.
screen.
Okay. काढ ना फोटो काढत राहा ना तुला चेअर घेऊन बसतात ना
Dr. Sandhya, I am sharing your presentation. Please proceed with your presentation.
Okay, sir.
Please go ahead.
Myself, good morning. Myself, Dr. Sandhya Tambekar Vanzari. I am the Assistant Professor in Microbiology from Dhotebandhu Science College, Gondia. Today, I am presenting a research paper on the production dynamics of extracellular protease from the newly isolated Bacillus odc from the Lonar Lake, Buldana, Maharashtra State, India. Next. And this is the abstract for my paper. Next. So, Lonar Lake is an impact crater that is situated in the Buldana district of Maharashtra state. It is a circular lake occupied by the saline water, and it is formed by the meteorite impact on the basaltic rock and well known as an inland saline lake with a considerable amount of sodium carbonate and chloride. It is a treasure of the extracellular enzyme that is amylase, lipase, protease, and cellulose producing Bacillus series. That is producing from the various types of the Bacillus species from which is isolated from the water and the sediments of the alkaline Lonar Lake. Next. These proteases are industry imported enzymes and used in the detergent, food and pharmaceutical and leather industry. This enzyme has the better resistance to the alkali and some other denaturing chemicals in the reaction mixture, having higher affinity toward the proteinaceous substrates. Next. So what is the aim and objective? As the alkaline, as I said, alkaline proteases produced by the Bacillus species are of great importance in detergent industry due to their high thermostability and pH stability. So very less studies has been done on the protease of the Bacillus from the Lonar Lake, which can be withstand at a high temperature and at a high pH, and has a wide application in the different industry. So as there is a large demand of the protease, isolation and production of protease enzyme is the most important to fulfill the demand. The present study aims to deals with the isolation, purification, characterization, optimization of the production of the protease from the Bacillus species isolated from the Lonar Lake. Next. This is the media composition which are used for the isolation of the halophilic bacillus from the Lonar Lake. This composition this composition is called as Horikoshi medium, which is taken from the Horikoshi paper, that is 1999, and this is made up of the various types of the component, that is glucose, starch, peptone, yeast extract, beef extract, magnesium sulfate, sodium carbonate, NaCl, and other ingredients, and this contain the various types of the medium A, medium B, and medium C and D. That particular 4 types of the media with the various constituents should be used for the isolation of the bacteria. Next. So this is the material method for the my experiment. Next. So in the present study, total 104 bacteria isolates are isolated from and maintained in the slant medium C, where which can I mentioned in the previous slide. And these various tests were performed by the identification of bacteria. And this inoculated alkaline skim milk agar at a pH 10.5 for studying the proteolytic activity and using the morphological and biochemical characteristics. Out of 104 cultures, only 37 isolates were identified as a Bacillus species, and out of 37 isolates, only 8 isolates were efficient in protease production. And most efficient protease production of Bacillus species are used for the as a preparation of the Bacillus species. So this Bacillus species comprises various types of the Bacillus, that is Bacillus horykoshi, Bacillus cereus, Bacillus fusiformis, Bacillus cassi, pseudoformis, and most importantly, next please. Out of them, most efficient Bacillus odyssey was used for the further study. This alkali protease production has the maximum pH at a 10, and this maximum production is recorded at the 72 hours. In the effect of substrate concentration, enzyme activity protease of Michaelis Menten constant and Vmax was found to be 4.16 milligram per ml, and the Vmax is found to be the 0.076 milligram per ml by linear Berg plot. Next. Next.
Ma'am, you are left only with the 1 minute.
Yes, sir. So this is the effect of substrate concentration, which are 4.16 milligram per ml. Next. This is the pH, no, that is a 10 pH, that is alkaline pH, optimal activity should be at the 10 pH. Next. The temperature that should be optimum at the 75 degrees centigrade. Next. So the carbon sources, that is a nutritional requirement which is used for the particular optimum activity of the protease. So here we show the lactose is the most efficient carbon sources for the production of the protease from the Bacillus odyssey. Next. The nitrogen, most efficient nitrogen is the soybean cake, and from the amino acid, the glycine is the most efficient of the nutritional requirement for the protease production of the Bacillus odyssey. Next. The metal ion, which is act as a cofactor, the magnesium ion, that is particularly act as a more efficient for the protease production. Next. So this is the compatibility. Previous. So this is compatible with the various detergent products, where we can see that the sorghum plus and the rein has a more compatible with this protease enzyme. Next. The isolated conclusion: the isolated Bacillus odyssey species from the Lona lake produces alkaline protease maximum growth at a pH 9.5, and so it is a very which was a thermophilic, alkalophilic, and has potential to produce good quality of the protease which can be used in the industry. Next. The Bacillus odyssey species were most efficient for the protease for the pH 10.5 at that is 75 degrees centigrade, and therefore it is operated at a high temperature, high salt concentration, and tolerate at the other environmental condition. This bacteria species is ubiquitous, it is non-pathogenic, not causing any diseases to the human being, and most efficient for the protease production among all the isolated protease producing bacteria. This addition to the limited number of the reports, Bacillus odyssey are the rarest rare bacterium was reported from the Lona crater sediments, and this finding has importance because it was the first isolated from the Mars Odyssey spacecraft in the 2004. Next. It was the first living bacterial biologic evidence of the origin of the Lona crater. Existence of this bacterium now rule out the possibilities of formation of the lunar lake by the volcano. And this bacterium was resistant to 100% of desiccation. Considering this desiccation property is in the gene, had now opened for the door of the biotechnology. All these properties of this bacterium were important in the changing global warming scenario, and in the occurrence of this bacterium will evolutionary in the future agricultural practices for the food security. Next.
Ma'am, could you please conclude? You have already taken 2 minutes extra.
Yes. Thank you.
The topic is open for the discussion now. What is the half-life? We have Dr. Ravikiran Pagare, sir, as a panelist. Is there any question from you, sir? Sir, you are on board with us, Ravikiran, sir. Please ask the question. Unmute yourself. Please unmute yourself, Dr. Ravikiran. Oh, he's in the studio.
Hello.
Yes, sir, we can hear you. Please go ahead with your question.
Yeah, madam, you told about that that is it is very rare species and it is found only in.
Sir, your voice is not clear, Pagari, sir. And the ma'am has already left the studio without answering the question. So we will proceed with the next presentation. Nipunage Bhagyashree.
Yes, sir.
Ma'am, we will start with your presentation.
Yes.
Please take a note of your time. You have only 6 minutes to complete your presentation. Ensure you have the noiseless background with you. I am sharing your presentation on the screen. Can you see your presentation, ma'am?
Sir, slightly zoom it.
Is it okay now?
Yeah.
Please go ahead. Your time
Thank
starts
you.
now.
Thank you. Good morning, everyone. Topic of my presentation is bioelectricity generation from different types of waste using microbial fuel cell. First introduction, energy demand is increasing day by day. So use of microbial fuel cell to generate electricity is a clean, reliable and efficient process. Materials and methods. In this study, we use 4 different samples. First is Mosambi peel, second is a pineapple peel, third is a zendu petals and fourth is a cow dung. Then we carried out enrichment and isolation. For this, we use sterile nutrient agar and sterile sample media. Then we check pH and temperature tolerance of isolates. For this, we use pH such as 579 and temperature such as 30, 37 and 60. Next point is a bioelectricity generation from different types of waste. For construction of microbial fuel cell, we use 2 plastic containers. Then in anode, we added 50 gram of crust sample, 100 ml of bacterial consortia and some amount of water. In cathode, we use 100 ml of
added 100 ml of distilled water and then boil produce for measure with the multimeter. Next point is screening for indole acetic acid. For this, we use Salkovsky's reagent test. Next point is results for screening of indole acetic acid. In this, all the isolate showed production of IAA. Isolate M3 showed maximum IAA production. Next is result for pH and temperature tolerance. In this, all isolate showed growth at pH such as 579 and temperature 30, 37 and 60. Results for bioelectricity generation. In for first is sample is Mozambique pills with consortium. At initial we got reading 0.202 volt. After 2 hours we got reading 0.248 volt, and after 4 hours we got 0.268 volt. Next is without consortium. At initial we got 0.121 volt. After 2 hours we got 0.220 volt, and after 4 hours we got 0.236 volt. Next sample is a pineapple pills. A result for with consortium. At initial we got 0.210 volt. After 2 hours we got reading 0.303 volt, and after 4 hours we got reading 0.306 volt. Next result for from pineapple pills without consortium. At initial we got reading 0.142 volt. After 2 hours we got reading 0.291 volt, and after 4 hours we got reading 0.30320 volt. Next sample is Zendu petals. Result for with consortium. At initial we got reading 0.14 volt. After 2 hours we got reading 0.18 volt, and after 4 hours we got reading 0.19 volt. Next part is for result without consortium. At initial we got reading 0.100 volt. After 2 hours we got reading 0.117 volt, and after 4 hours we got reading 0.184 volt. Next sample is a cow dung. Result for with consortium. At initial we got reading 0.037 volt. After 2 hours we got reading 0.072 volt, and after 4 hours we got reading 0.120 volt. Next for without consortium. At initial we got reading 0.302 volt. After 2 hours we got reading 0.364 volt, and after 4 hours we got reading 0.382 volt. These are all results for bioelectricity, and all results are in the volt. Next part is a conclusion. This present study, from this present study, we can conclude that all 13 isolate obtained from 4 different samples, that is Mozambique pills, pineapple pills, Zendu petals, and cow dung, have ability to generate bioelectricity using microbial fuel cell. All isolate showed application in form of production of indole acetic acid. This indicates that isolate could enhance root and shoot development of plant. All isolates showed growth at pH such as 579 and temperature such as 30, 37 and 60 degrees Celsius. This can conclude that our project is not restricted to only certain pH ranges and certain temperature ranges. It can be carried out at pH such as 579, and if it is successfully showed bioelectricity production at temperature such as 30, 37 and 60 degrees Celsius. Thank you all of you.
Thank you, ma'am.
Thank you, sir.
We will just put some questions. Is there any questions from the audience side? So there are no more questions from the audience side. Thank you very much. Thank you, ma'am.
Thank you, sir.
The next presenter with us is Manjusha Sonole. Good afternoon, ma'am.
Yes, sir.
Good afternoon.
Good afternoon, sir.
So you are on board with us to present your PPT. I will be sharing your presentation. Once it is visible on your screen, please proceed for the presentation.
Okay.
Ma'am, your presentation will be visible in 5 seconds. Please proceed.
Okay. Thank you. Sir, can I start? Hello. Sir, can I start?
Yes, please go ahead.
Okay, thank you. Myself, Manju Samule. I am from Swami Ramananda Tirtha Marathwada University, Nanded, and my topic is the extraction and characterization of algal oil from the Cara vulgaris. I have choose this topic because nowadays there are so many problems are belong to for the production of biofuels. There are so many people are using as a main type of plants and animal oil for the production of algal oil, but I have choose the algal species because nowadays there are so many species of algae are available in the watery medium, and out of that I have choose that is the Cara vulgaris. And depending on these all things, I have choose the aims and objective, that is the first objective of my study, that is the taxonomical identification and extraction of algal oil, TLC analysis and GCMS analysis. Um, the morphological characteristics of the Cara vulgaris, that is a it is a green in color, belongs from the class Chlorophyceae, and it was collected from the Kidney region and Bamni region, and it was identified with the help of monograph of Piscott. And this species is mainly collected from the Eldari Dam area from the Parbhani district, and it is one of the most largest dam from the Parbhani district. And after the collection of algal sample, it was shed dried, and the extraction of algal was done in the laboratory with the help of Stocksless method by using the by using the solvent, that is the hexane. Next, after the extraction of algal oil, it was calculated with the help. Sir, next slide. Sir, next slide. Huh? After the extraction of algal oil, it was calculated by volume method, and that is the percentage of total oil contained in the algae was calculated, that is 32%, and the pH was measured, that is 6. And after the extraction of algal oil, it was performed by the process of transesterification process, and that algal oil is collected in the form of crude algal oil, and that crude algal oil was performed with the thin layer chromatography. After the thin layer chromatography, the as per the RF value, it was calculated that presence of cardiolipin and lauric acid in the respective algal soil. Next slide. Sir, next slide. After the thin layer chromatography, the GCMS analysis was take place. Uh, GCMS analysis was take place in the safe from Mumbai, and depending on their data, there are total 9 compound was identified at the different molecular weight with differentiation time and different molecular formula, in which the presence of 2-propanic acid, axobis, oleic acid, 3, 7, 11, 15 tetramethyl, 1, 2 hexadecone, pentadecanon, then panthalic acid, butyl tetralic acid, hexonic acid, N hexadonic acid, phytol, and 1, 2 benzodectanoic acid, diasputase. So these total compounds was identified, and depending on this, all the data, it was concluded that the presence of unsaturated and saturated fatty acids shows that this algae is very useful for the production of biofuel. So its end.
Thank you, ma'am. So we'll be forwarding you the questions raised by the audience. Ma'am, you can see the question on your screen. The question is raised by, why are you not using the other species of algae?
Easy. Sir, I have used the other species also, but I have only show the only one species.
Okay, thank you. The question is, why only this species? Could you please elaborate the answer? Why only this species?
Why species? Okay, sir. In that area, there is a large amount of this species was found in the rich amount, and basic thing is that it is found near to the farming area in a large amount. So I choose in this algae because it's available in large quantity and naturally in the environment.
Okay, we have got one more question.
Okay.
What are the tools used to characterize the compound? The
What
question
are the
is
tools used to
characterize the compound. The question is, on your displayed on your screen. What are the tools used to characterize the compounds?
These compounds are identified by using literature.
Okay, fine. We have the last question with us. What is the biodiesel conversion efficiency of this algal oil? Biodiesel conversion efficiency. What is the
Sir, your
biodiesel?
sound is breaking.
Hello, can
Sound
you
is
hear me?
breaking. Yes.
What is the biodiesel conversion efficiency of this algal oil?
Sorry, sir.
I repeat the question. Biodiesel conversion efficiency of this algal oil.
Sorry, sir.
Okay. Thank you, ma'am. Thank you for your nice presentation. Thank you. We have the next presenter with us, Sonone Chandrakala Ganesh Rao, but she is not connected. Probably her microphone and cam is not working. Ma'am, if you can hear us, please ensure that your microphone is working. Only then we can take you on board to the studio. Voice not connected. Now we will go ahead with the next presentation. Professor Hatindran Prakash. Hatindran Prakash Jadhav. Sir, could you please unmute yourself? We can't hear you. You are not audible, sir. You are not audible, sir. We will proceed with the other presentation. Meanwhile, you can check. We will provide you the another slot at the end of the other presentations today itself. You kindly verify your microphone and other things. We have the next presentation. It is Dr. Omar. Professor, could you please unmute yourself? You are not audible. Dr. Omar, we can't hear you.
Okay, I'm on.
Yes. Thank you. Thank
Hello?
you, sir. Hello,
Thank
can you
you,
hear
sir.
me, sir? Yes. I'll be
Yes,
sharing your
I can
PPT.
hear you.
I'll be sharing your PPT. You have 6 minutes of time. Kindly complete your presentation in 5 minutes, followed by 1 minute discussion.
Okay.
Once the PPT is visible on your screen, Kindly proceed for the presentation.
Okay. Okay. Hello.
Sir, please proceed for the presentation.
Thank you. Thank you. Good afternoon. It's glad to have me here and to have you there. My name is Umar Aliyu Hangwa from the Department of Pharmaceutical and Medicinal Chemistry, Ahmadu Bello University, Zaria, Nigeria. My topic of this afternoon presentation is the isolations of quercetin 3-O-beta D glycopyranosyl 1,6-alpha rhamnoside from the acetone fractions of the stem bark of Stereospanus kunthianum. Next, please. Next. Plant has been with man for ages. From the ancient time, man has been using plant for one thing or the other. The medicine man, probably an accomplished botanist, represents probably the oldest professional man in social evolution. Through the process of studying nature, early man observed consistency in the chains and patterns of natural phenomena. This was the empirical acquisitions of knowledge. In this context, man studies plant, some of which he found to be food he cultivated thus, some of which were harmful or toxic. Then he rejected this category, and yet another group he found to be medicinal, they contain medicinal properties. All these categories he translated and extended the knowledge to his progeny. Primitive society believes in healing by similarities. For example, red resins is an example of a plant that was used for the blood. A heart-shaped leaf signals its cardiac properties, while a liver-shaped leaf was a sign of efficacy against jaundice. Plant, with its numerous metabolites, has served man in so many ways, to the extent that researches are being carried out on this plant. It is our effort to add to the database of the chemicals that are gotten from the plant. Thus, we investigated the plant Stereospanum kunthianum. Next, please. Next, please. The plant Stereospanum kunthianum is a small or medium tree, 15 to 20 meter or less. It is cut across the Sudan-Guinea savanna and the Sahel region of Africa. From the map there, we can see right from Chad, cutting across Nigeria, Burkina Faso, Mali, Senegal, part of Niger. All these are areas where we can find this plant, Stereospanum kunthianum, that belongs to the family Bignoniaceae. It is naturally found in dry areas of deciduous forest, mountain, termite mounds, rocky outcrops, and windland and near streams. Part of its ethnomedicinal uses, it is used in the treatment of ulcer, skin eruption, leprosy, pneumonia, gastritis, diarrhea, gonorrhea, hypertension, inducing labor, and the rest. Chemicals so far isolated from these plants, a bioguided fractionations of the Stereospanum kunthianum root extract led to the isolations of 4 noble Kunthianums. Enagi et al. 2002. Also, one erudite and two phenyl propanoid were isolated from the plant by Faladon et al. 2009. Epilamide was reported or isolated in the stem root back of the acetone fractions of Stereospanum kunthianum by Hanwa et al. 2015. Next, please. This is the natural habitats of the plant. You can see that it is a rocky area and the growth is retarded by the nature of the land there. Next, please. The plant Stereospanum kunthianum fresh was collected and was taken to the herbarium sections of the Department of Biological Science, Ahmadu Bello University, Zaria, Nigeria, for identification and authentication. It was identified by a botanist by comparing with an existing specimen with a voucher number. The plant, the stem back was prepared and dry under shade, reduced into powder using mortar and pestle. We've gotten 2 grams of the powdered plant materials, which we soaked into methanol for cold maceration for 11 days with constant shaking and agitation, thereby affording a black oily substance coded at methanol extract crude. This crude extract, part of it was used to screen the metabolite, the constituents present in this extract, following a standard protocols. Silva et al. 1998, Tris and Evans, 1982, Toner and Brain and Toner, 1975. 20 grams of this crude extract was partitioned successfully using hexane, ethyl acetate, and acetone. The acetone fraction, whereby we've gotten the hexane extract, ethyl acetate extract, and acetone fraction. The 10 grams of the acetone fraction was mounted on a glass column over silica gel and eluted with pet ether and a mixture of pet ether, chloroform and ethyl acetate, and methanol. Elutions were monitored through use of TLC. Total of 190 fractions were has been collected. So thereby, those with the same RF value were pooled together.
Excuse me, Professor. Professor, please conclude. Please conclude.
Okay, sir. So in this, we were able to isolate this from the eluent number 14 within the range of 80, 82, and we've gotten a brownish yellow substance that we've coded NH2 that was subjected to physiochemical analysis taking the melting point, the phenolic test using Shinoda test, and the ferric chloride test. The spectrals were analyzed using the UV, IR, and the 1 and 2D where NMR were also used to analyze the spectra. The result, please. Next. Okay, this is the result at which we've obtained the area where we've isolated our compound. Next, please. Okay, so using, can I have the spectra? Can I have, go to the spectra? This is the UV showing the two absorption band. Next, please. The IR showing the frequency of the hydroxyl carbonyl group, and the methanol group. Next, please. This is the NMR proton, the proton NMR of the spectrum of NH2. It is looking at this spectrum, you can see that it is compartmentalized. That is what it means. That is, it is divided into four. The downfield indicating those signals observed in the A ring of the flavonoid. At 7.64, we have the hydrogen doublets with J value 2.0 Hz. And next, we have another signal at 7.62, we have a proton doublet with 2J value calculated, 2.0 and 8.4. That, again, around that region, a signal at 6.88, we have a proton doublet with another J value of 8.4. This means that the 6.2, okay. Next, please.
Professor, please
Next,
conclude
please.
your. Professor, please
Okay.
conclude.
Okay.
Please conclude.
Okay.
We're almost already
This is
3 minutes.
This is the carbon 13 showing the 27 carbon in the compound. Next, please. The depth indicating 1 methane, 1 methyl, and the rest 15 as methane compound carbons. Next, please. This is the COSI where the protons correlations were observed. Next, please. So this is, after taking those spectrum into considerations, we compare with the existing structure where we compared our findings with the findings of Gant et al. 1998, Wundt et al. 2007, and Mexia et al. 2008. Next, please. So with this put together, we've come to the conclusion that the compound is routine, as it is trivial name. Chemically, it has a number of pharmacological activities, including antioxidants, cytoprotective, vasoprotective, anticarcinogenic, neuroprotective, and cardiovascular. Thank you for listening.
Thank you, sir. We will take the questions now.
Yes, sir.
So the question is visible on your screen.
Okay.
What is the application of this product that is obtained by the study? Application of your compound isolated.
Okay. I have just mentioned in the conclusion that it has so many pharmacological activities. It can be used in the treatment of cancer, in the treatment of a cardioprotective, as I mentioned in the conclusion.
Okay, thank you. The last question is, what is the yield difference between the oxalate extraction and the cold extraction? You have done the cold extraction rather than going with the oxalate extraction. The question is from Dr. Aitekar.
Yes, we are targeting a particular group of metabolite. So we feel that using cold maceration will give us a good a good contact with the phenolic compound, which is of our interest. That is why we didn't go for oxalate extraction.
Thank you, sir. Thank you very much.
You're welcome, sir.
Thank you. Thank you, sir.
Thank you, sir.
Give a student is going to come now. The next presentation is Vaishnavi Harish Anil. Due to some technical issues, she is unlikely to join us now. So we'll switch over to the next participant, Ashwini Bosle from Sandip University. Ashwini, could you please unmute?
Yes, sir.
Ashwini? Okay.
Yes, sir.
Fine. So you kindly start as soon as your PPT is visible on your screen. I will be sharing a
Okay.
screen with you now.
Yes, sir.
Please proceed with the presentation, Ashwini.
So can I start, sir?
Yes, yes, please go ahead.
Okay. Am I audible?
Yes, go ahead. Your time starts now.
Okay. Okay, sir. Okay. Hello, everyone. I'm Ashwini Bhosle, pursuing my master's at Sandip University. I'm representing my poster, which is a survey report carried by myself on the topic kidney and urinary tract infection with respect to water intake and working schedule of people. The aim to study current practice among people of various professions in Nashik city regards to investigational management of
Ashwini, we have lost the connection with you. Ashwani, can you hear me? We have lost the connection with you. You are not audible.
Hello, am I audible?
Yes, now you got disconnected. I'm sharing the screen again. Huh? Please
Okay.
ensure you are connected. Huh? Go ahead with the
Yes.
presentation.
Okay. Hello everyone, I'm Ashwini Bhosle pursuing my master's at Sandip University. I'm representing my poster, which is a survey report carried by myself on the topic kidney and urinary tract infection with respect to water intake and working schedule of peoples. The aim of study current practices among people of various professions in Nashik city regards to investigational management of urinary tract infection and kidney disorder with respect to water intake and it's important in their daily busy schedule. Next, please. Next slide. So here 113 participants from same health region were participated in this survey. So the survey concluded that there are totally 113 participants from which 89% respondents are belong to 20 to 25 years of age group and remaining 5.5% are more than 25 years age group, from which 57% respondents are female and 43% were male. Occupationally 77.5% are student, 14% are employee, 3% are businessmen, and other belongs to the various categories. And their working schedule, working hours per day are 40% of peoples work 6 hours per day, 37% work 8 hours per day, 11% work 12 hours per day, and 10% work 10 hours per day, and their water intake, daily intake of water is 35% takes more than 3 liters of water. 34% take intake is less than 3 liters, 23% take less than 2 liter of water per day, and 8% drink less than 1 liter water. The aim to take questions like this, like occupation, working hours, and their water intake, is just to reveal their habits of drinking habits and their working schedule. Next is, from these respondents, that is 113 respondents, 10% of the people suffer from kidney disorder and 15% suffer from urinary tract infection. Majority of the type of infection seen is acute infection, which is 73% people suffer from acute infection, and 27% suffer from chronic infection. From all participants, 83% have knowledge about relationship of water intake and urinary tract disorder or kidney disorder. And from them, 69% people avoid the use of urination or washroom due to their workload. 81% people agree that busy schedule affect the health. The conclusion of my survey is just to it is just a preliminary survey. This survey reveals that the water is an important factor to maintain kidney and urinary tract health. To study the infection rate of kidney disorder and urinary tract infection is necessary because many students and employees hesitate to share their problems due to their busy schedule, and it sometimes lead to avoid urination, which may cause serious health issues like kidney stone or kidney problems, and may cause urinary tract infection in females. It is suggested that the monitoring of daily consumption of water will be helpful to avoid UTI or any kidney disorders. Thank you so much. Hello.
Thank you, Ashwani. Thank you, Ashwani, for the nice presentation. We'll go to
Thank you so much, sir.
uh, for the questions. Please be online with us
Yes, sir.
to
Yes.
answer the question. Thank you, Ashwani. There are no questions for you. Thank you.
Okay, sir. Thank you so much.
मैं हेमेंद्र को ले लूंगा, उबले है, विष्णु हरीश अनिल नहीं. I will take Professor Jadhav again. Professor Jadhav, Professor Hatendra Prakash Jadhav. Can you hear me? There is still a problem with your audio. You are not audible at all. Yes, can you say something, Mr. Jadhav? Sorry, we can't hear you. Please come again, verify the microphone. Thank you. We will take another presenter on board. Hello, good afternoon, Mr. Obale.
Hello, sir. Good afternoon. Good afternoon to all of you.
Good afternoon. I will be sharing the presentation with you. As soon as
Yes,
it
sir.
gets displayed on your screen, kindly proceed with your presentation.
Okay, sir.
I am about to share. Kindly go ahead with your presentation.
Yes, sir.
Please proceed.
So good afternoon to all of you. I am here to inform all of you that honey's importance and honey's antibacterial importance. Honey is the sweet substances produced by the Apis mellifera, which is an insect which collects the honey from the blossoms of the new flowers and collect it in the honeycomb, and then by their some after some days these blossoms nectar converted into honey. Honey has medicinal value as it contains antimicrobial properties. Honey is also used for the wound infections and bacterial infections. Wound is caused by yes sir. Wound is caused by some laceration or the accidental damages. Laceration is the deep cut to the skin. Wound goes further goes to the hypoxia, which is prevented by honey. Therefore, in Asian countries, honey is used as the wound inflammatory infections. Honey also has antioxidant properties. It takes to the osmosis in the blood. Honey is directly entered in the blood system without digestion, which further leads to the osmosis procedure, and it is useful for the diabetics and typhoid like diseases. Honey is effective against. Hello, sir.
Yes, please go ahead.
Hello.
Conclude your presentation, please.
Okay, sir. Honey is also used for the diabetics, and honey some scientists noticed that honey is effective against the viruses, but it has no any medical proof to prove that honey is effective or not effective to the viral infections. Yes, sir, I have done.
Thank you very much for your presentation.
Thank you, sir, all of you, and thanks.
The next presentation is Dr. Abdul Jalal. Next is Dr. Asha Bahusab Kadam. Unfortunately, she has not joined us yet. The next presenter is Chetali Ambadre. The next presenter is Hima Wadera.
Good afternoon, all of you. Am I audible?
Yes, you're audible, but your cam is still off. Could you please put on the camera?
Okay.
Yes. Welcome, ma'am. I will be sharing
Thank
your
you.
PPT. I will be sharing your
Okay, sir.
PPT. As soon as it is
Thank
visible to you, kindly proceed with the presentation.
Okay, sir. Today's my topic is qualitative
Wait,
character.
just a minute. I
Okay.
have to share your presentation.
Okay. May I start?
Yes.
Okay. Today's my
Please
topic
go
is
ahead.
qualitative characterization. Okay. Today's my topic is qualitative characterization of phytochemical presence in bark of some tree species. The main aspect to choose this topic is due to the we are use many tree species and their plants in our day as a medicine. The plant have been choose are Bohemia purpurea, Calendra hematocephala, Dalbergia sisu, Deris indica, and Peltophorum pterocarpum. These plants are commonly found nearby around us as a garden tree or as well. They are also reported as their use in as a medicine as traditional medicine in Ayurveda. The 5 tree species are belongs to order Rosales. The member of this order are noted medicinally importance like Acacia species, etc. The plant which we include in the this project are common use like anti-inflammatory antioxidants. They are reported as a anti-malarial property as well. The main aspect is to to prepare a identification tool with the help of phytochemical database. It could help in identify tree species in the absence of their primary structures like leaves, flowers and fruits, and as well they are used for medicinal purpose. In the absence of this primary structure, we can identify the proper plant with their bark patterns or the phytochemicals are present in it. So I have include the result. There is a I have considered the qualitative analysis like test for carbohydrates, proteins, phenols, tannins, flavonoids that can be used as a path to lead to us to the medicinal purpose of the plants. We can see that the plant which are all plant in contain reducing sugars like calendra and phytochemical peltophorum consider monosaccharides. They also consider the amount of tannins and phenols which can be used as antioxidants, and as well they are also shows them properties like anti-inflammatory, anti-malarial, and used also in dysentery as a memory improver. This is the main aspect of my poster or my work is to conclude that if we consider a bark as an identifiable tool and for the medicinal purpose, we can include them as a uh, um, modern medicine, their component, and we can use as a medicine in a raw or a decoction form, so it cannot be harmful as well as their allopathy medicines. So that is my work. Thank you so much. Am I audible?
Thank you, Hima. Thank you for the nice
Hello.
presentation. We'll go
Okay,
ahead.
thank you, sir.
We'll go ahead with the questions.
Okay, thank you, sir.
So we have a question from
Okay,
Mr.
sir.
Nilesh Walke. The first question
Yes, sir.
is, have you done any quantification of alkaloids? You have done the testing,
No,
qualitative testing.
only I have this is the first step. That's why I'm doing only the qualitative analysis, and further I can consider the nutrition as well as the quantification of these phytochemical compounds.
Okay. Thank you, madam, for nice presentation. Thank you.
Thank you, sir. Thank you, sir.
We have the next presenter with us. Dr. Abdul Jalal from Nigeria. Hello, Professor. Am I audible to you?
Hello. Good afternoon, members and participants.
Good afternoon. Good afternoon, sir. I am sharing your
Yes,
presentation from my end. As soon as it is visible to you, kindly proceed for the presentation.
thank you very much. My presentation is on level of chromium, iron, and zinc in selected Nigerian foods and vegetables using neutron activation analysis. Now, the neutron activation analysis is a method that does not involve chemical treatment and is using the nuclear reactor from the Ahmadu Bello University. Now, essentially, the chromium, iron, and zinc are essential elements that are required by our body, and sometimes their deficiencies need to be supplemented. And these ions of chromium, iron, and zinc, they appear in their combined state of oxidation state. That is, they have the +3 for chromium because it has two oxidation states, +3 and +6, but the essential one is +3, and then iron 2 instead of iron 3, and then zinc 2. So these are very important nutrients, and sometimes their deficiencies has to be supplemented. Next, please. Now, most importantly, the zinc itself, it has a very important role in the system because it protects the structure and the integrity of the protein, and therefore it regulates the gene expression. And that is why in the present COVID-19, zinc tablet is essentially recommended combined with chloroquine hydrochloride, in the sense that the chloroquine being used as a mobility, but the zinc itself prevents mutation of the cell, therefore of the protein. It prevents the coronavirus from multiplying, and that is what is meant by gene expression. So it protects that. So that is why it's used in the present and treatment. And its deficiency, including in infants, is usually very devastating, and care has to be taken. Next, please. Next slide. Now, essentially, the items, the common items, maybe they are all the same, still found in India. You have the Irish potato, the lettuce, the cabbage, and then the rice and all other things there. So, gotten from a local market there in Zaria, Kaduna State. Please go ahead. Next slide. Now, the most important thing is the method. This method does not involve chemical treatment. It is just the raw foods that will be treated, and that after the treatment and the removal of all the dirts, these materials are now grinded and are now subjected to the nuclear reactor. The NIRR-1 Nigerian research reactor was used. It is a low-powered reactor, and you know that it uses uranium-235 isotope as the fuel. Therefore, what happens there essentially is that it releases neutrons. These fast-moving neutrons normally have non-elastic collision with these food items or vegetable items subjected to this bombardment. As a result of this bombardment, there will be excitation potential of energy in these materials. And then after the excitement, then this excitement will now decline. And that is when the counting starts. There are two sets of countings. In fact, the irradiation is of two types. There is what we call the long irradiation and the short irradiation. And the categories of these foods belong to long irradiation. So the nuclear bombardment is for 6 hours, and then there the counting begins. It is from the counting now that you find out the decay constant. This decay constant is used in calculating the half-life of these materials, and eventually gives you the concentration of each of these nutrients, chromium, iron, and zinc in these food items. So please let's go to the results. Next slide. So you can see there that the half-life, you can calculate using 0.693 divided by lambda. The lambda is the decay constant, which gives you the calculation to give you eventually the concentration of the ions present. Please, next slide. Next one. We use some method of standardization. The standardization is a relative standardization. We normally combine both the material that we are taking for the experiment and then the standard existing material together. We put them in the reactor and subject them to this irradiation for 6 hours. So that is what we call relative standard method. Please, next one. Go to the results. The next result. So we can see clearly from the bar chart there, this one indicating the fruits and vegetables. The presence of iron there is very high in maize. You find that maize has a significant quantity in iron, and the lowest there it has also is in rice. So and iron is very important in terms of in our body, in terms of the clotting of the blood and so on. So these are all the various concentrations of the food items. The least is rice and the highest there is the maize. Please, next result. Now this is the table for the vegetables. Please, the bar chart. Now you discover that you can essentially see the long bars, but the ones that are very short, narrow towards the base are usually essential nutrients like chromium, which comes in a very small quantity, but it has the highest amount in lettuce and the least in rice. And you find that about the iron. The amaranthus, that is the leaves, the spinach leaves has the highest in iron. It gives maximum in iron. And also the zinc, we find it very much available in maize. So you find that in the lettuce, the green vegetables generally have high concentration of iron, and therefore they are very essential. However, their consumption must be controlled in order not to have an effect on the metabolism of vitamin D. Next, please, go to discussion. Now, essentially, all the ions, chromium, iron, and zinc were all found in various concentration in all the food and vegetable sample. So that is why the foods in the found in the third world countries, which does not undergo so much secondary processing, is essentially very good and healthy. But with the over processing, we normally lose most of the nutrients and eventually eat something that more or less like a garbage. So care must be taken. In all, the concentration of these ions are within the acceptable limit of the World Health Organization, the Food and Agricultural Organization, and the European Food Safety Association. You discover that except, of course, for iron, in some food items there, that is slightly above the standard, the World Health Organization standard, which some control measures has to be taken, and we have to take seriously into consideration the dietary intake per day. You find that for some it will be around 8 for females especially, and then for 12 for male. So these kind of thing must be taken into consideration in order not to have overloading. The overloading of it affects the metabolism of vitamin D. Please, next. Now, the presence of all these ions in our body, which is very essential, as well as utilizing them without undergoing much process. Now, this is to say that there is the likely change in the concentration of these ions when it is subjected to cooking, but we are trying to find the raw concentration in its original form, and other plus or minus in terms of these ingredients might be experienced when you eventually analyze the cooked foods from this. So it's within the reach of the World Health Organization of 250 milligram per day, or the European Food Safety Association of 0.5 milligram per day for chromium. And chromium is very essential, particularly in insulin sensitivity, because it potentiates the production of insulin, therefore is very essential for diabetic patients, particularly those suffering from diabetes mellitus type 1, which is not producing enough insulin. So chromium has a very important role there. So in conclusion, we have discovered that all these items, they have these ingredients, so therefore it's better we keep them in our diet, and dietitians should be able to recommend them for diabetic patients. And all in all, you will discover that rice has some of these least ingredients. Perhaps that is one of the reasons why in most treatment of diabetes, consumption of rice is advised to be put to the minimal. So this has given us an idea of the food at our door mouth, the food at our backyard, the essential ingredients. And we have taken the neutron activation analysis technique using the nuclear reactor to find the original concentration before they are subjected to treatment for eventual consumption. I thank you very much for listening.
Thank you, sir, for this invaluable presentation. We will
Thank you.
take the questions from the audience.
Okay.
Sir, the question is visible on your screen. Could you please answer? Please give some other examples of chromium-containing vegetables.
Yes, the green generally have this. We have lettuce, we have cabbage, we have amaranthus, we have the Irish potato, we have the spinach, and we have the carrots. All these were analyzed, and as I've mentioned, that all the three components, including chromium, they are very much available. But essentially, it has the highest content in lettuce.
Thank you, sir.
Thank you, Prof.
We will take the another question. Sir, we have one more question from Dr. Raitika. Could you please explain the extra source of chromium so that we can increase its concentration in the food?
Sorry? The extra source?
Source of chromium to increase its concentration in the
source
food.
of chromium.
Yes.
You know, it's an essential ion. So it appears it's a micronutrient. It does not appear in large concentration. Besides, there is also the standard control of consumption. For example, by the World Health Organization, it is 250 milligram per day. That is on dietary consumption. So no matter the concentration, you should not try to go above the limit of dietary consumption per day. 250 microgram per day for the European Food and Food Security Association, 0.5 milligram per day and for the FFO 10 milligram per day, 10 milligram for the male and the 12 milligram. So we must try to maintain within the limits of the World Health Organization for safety, because sometimes if they are in excess, but it's very difficult among the ions for chromium to be in excess, because it's coming as a micronutrient and it potentiates the muscles in the stimulation of the sensitivity of insulin. So the greens are essentially the main content that contain a lot of chromium. As I mentioned, lettuce is among the leading ones.
Professor, we have one more question from Dheeraj Shekhawat. What is the duration of short-time irradiation? The question is visible on your screen.
What is the duration of the short-term radiation?
Yes, sir.
The short-term radiations takes about 2 hours, but the long-term takes 6 hours. So there's what we call the delayed counting, and this delayed counting, as I've told you, is as a result of de-excitation after the non-elastic collision during the radiation of the excitation. After that, then you wait. There is the one from 5 to 10 days, and then there is also a waiting period, especially for the longer radiation, from 10 to 15 days. Within this, it will tell you the half-life, how much of the contents that will finally be evaluated from the decay constant to show you the concentration of such things. So it's essentially 2 hours, and then for the longer irradiation, 6 hours. And the waiting period differs from 5 to 10 days, and then for the longer one, from 10 to 15 days.
Sir, the extra question is there visible on the screen. Could you please answer this?
Okay, any studies?
Studies on side effects of irradiation. Side effects.
Well, the irradiation is essentially a technique, and there is, of course, but there are all preventive measures when you go into the nuclear reactor, so that the radiation will have minimal effect, apart from the usual protocols. And you know that within the reactor itself, it has what you call the graphite, which absorbs the fast-moving free neutrons, which it does not irradiate to cause harm to you. And of course, you must avoid things like even your wristwatches that can easily be magnetized and so on. But there are standard protocols. You know that nuclear reaction is not something that you should handle very casual. So there is a standard protocol for carrying this, and there is no any risk. Besides, you are not the one being irradiated, it is the food item. Okay, so you are safe.
Thank you, sir. Sir, we will take the last question.
Okay, please.
Does the high concentration of iron pose any threat to the health? Please.
Yes, it does. Yes, it does. Care must be taken so that, as I mentioned earlier on, among all the three components, all of them are within the limit of the World Health Organization. But you discover that the iron in particular is very high. It gets to the extent of 450 milligram there present. So it's present. It is likely to cause what you call overloading, and this overloading sometimes can affect the metabolism of other ions, particularly the vitamin D. And you know that vitamin D is very important in terms of maintenance and strengthening of the bones, and then also it's a sort of kind of boost immunity. So if such things suppress because of the overloading, then there is likely to be some impairment. So yes, it should be controlled. There is what we call overloading there, particularly of iron of high concentration.
Thank you, sir. Thank you very much for this beautiful and informative presentation. Thanks a lot, sir.
Thank you very much, Prof. And thank you, my good audience.
Thank you, sir. Thanks a lot.
Thank you.
We will now proceed with the next presentation. Mr. Hatinder Jadhav, I will give you the last chance. Please. Dr. Hatinder Jadhav, you are on board. Could you please verify your microphone?
Yeah, is it audible now, sir?
Yes, yes, yes. Thank you very much. Thank you for
Yeah,
rectifying the
finally.
issue. I will be sharing with you your presentation. As soon as it is visible with you, kindly proceed for the presentation.
Sure. Shall I proceed?
Wait for the presentation to appear on your screen.
Okay, okay.
Please go ahead.
Yeah, hi. Good afternoon to one and all. The title of my today's topic is Statistical Based Bioprocess Designed for Enhanced Production of Alkaline Protease by Bacillus species, which we had isolated. Please, next slide, please. So as a challenge, we are facing a very big issue in agriculture sectors in which we are using a very harmful chemicals for the elimination of phytopathogens which are responsible for the various plant diseases and which are also responsible for the eradication of the agricultural soil. So to eliminate this problem, we are conventionally using the chemical fertilizers which are having a bad impacts on the soil and also on the yield. Next slide, please. Which we can see that these phytopathogens have various harmful impacts on the groundnuts. Next, please. Cereals, next vegetables and fruits also. So we can see that in all the sectors they are creating a damage. Next, please. These are the major issues which are related with the use of chemicals for the treatment of these phytopathogens impacts. Next, please. So as a biological solution, the plant growth promoting rhizobacteria was come up, and the term was coined by the Joseph Kopler in 1978. And these plant growth promoting rhizobacteria are the microflora which are living into the rhizosphere and which are able to do plant growth promotion with the various aspects like zinc solubilization, nitrogen fixation, phosphate solubilization, iron nutrition, and phytohormone production, as well as they provide the biocontrol to the different phytopathogens by the secretion of antibiotic, induced systemic resistance, competition, mycoparasitism, hydrogen cyanide production, and lytic enzyme production. So you can see these are the major advantages of using these biological solutions. The major one is it is not inducing any resistance into the phytopathogens, and we can use it repeatedly, and it also increases the productivity, and it is not having a harmful effects as a chemicals on agriculture sector. Next slide, please. So here in this research, we had narrated the topic onto the hydrolytic enzymes, as the hydrolytic enzymes are having various industrial aspects, which earlier one of the speaker already explained that extra, but here we had focused on the extracellular microbial enzymes, which are having a high advantages over the plants and animal sources. So these are the main, next slide please. These are the main basic hydrolytic enzymes. Next please. Yeah. So the chitinase, protease, cellulase, and glucanase are the various main hydrolytic enzymes which are used for the hydrolytic elimination of phytopathogens. Next please. Over these protease we had selected because it is having a broad range of working temperature from 10 to 18 degrees Celsius, while it has the ability to work into the broad range of pH also, which is 4 to 12, and which increases the demand of protease into various sectors like agro industries, pharmaceuticals, and food processing industries. Next, please. So with this, we had decided our objective is to isolate the microorganisms which are able to produce the protease and other hydrolytic enzymes, then the identification of these, and the statistical based optimization for the maximum production of these enzymes. Next, please. So we had selected the various different locations for the isolation of these isolates, you can see here, and after that we had used the minimal salt medium with the different carbon sources, so for the isolation of different uh, hydrolytic enzymes producing microbes. Next, please. So you can see in the picture we had used the chitinase medium for the isolation of chitinase producing isolate, and the zone of hydrolysis over the colony you can see is because of the chitinase activity on the chitinosome present into the medium. Next, please. This is the another chitinase producing isolate. Next, please. This is the proteolytic activity on the skim milk agar by the isolates, where we can see the due to the proteolysis of the protein present into the medium, the zone of clearance has appeared, which indicates the protease production by the isolates. Next, please. So this is the proteolytic activity. Next, please. This is the cellulose producing microorganism plates. Next, please. These around 120 isolates were isolated. Around that, 9 isolates were found to produce the multiple hydrolytic enzyme, which you can see this is at the center of the Venn diagram. And these isolates were identified using the polyphasic identification techniques, which include the 16S RNA sequencing, biolog, MALDI-TOF LMS, and GCFEM analysis. Next, please. On the basis of the results, we had identified these organisms as the Bacillus cereus, next please, Bacillus xanthelicus, next please, and Bacillus brevis species. All these isolates sequences were submitted to the gene bank, and the cultures were submitted to the NCCS Pune. Next, please. For the production of protease, next, please. For the production of protease, we had the minimum salt medium, which is composed with the minimum nutrients and for the protease production, and all these isolates were uh, subjected to the protease production. From that, we had got the best best protease producing, that is the maximum protease producing organism, that is Bacillus. Next, please, Bacillus cereus, and for that. Using the one variable at a time approach, we had studied the different physico chemical parameters individually on this production, where we had found that the incubation period of 30 degrees Celsius, 48 hours, incubation temperature of 30 degrees Celsius, inoculation concentration of 1 point. You can see the variables which are optimum for the maximum production. Next, please.
Please conclude.
So in yeah. Then to study the significant response by these variables onto the production, we had subjected these variables into the statistical model, and we can see that the next, please, next up to the Pareto chart. Yeah, again one more. Yeah, you can see here we had selected the 8 variables, and out of these 8 we have found that the 3 variables, which are the pH, carbon and nitrogen source, are found to be the significant for the protease production. Next, please. Yeah. Here into the CCD model, we had selected these the significant variable to understand their interactive role and individual role for on the protease production. Next, please. So 20 different sets of experiments we had designed, and in that we had found that, next please, next again. Yeah, so here previous one. Yeah, so the software has from these different experimental runs, software has predicted the significant concentrations for these significant variables and predicted the maximum yield. Next, please. So by using the statistical results, we had run the bioreactor, which is a Indian made, and using this value, we had taken the production. Next, please.
Professor, you are requested to conclude your presentation.
Yeah, it's done. Yeah, it's done. So we can see that in unoptimized media and into the optimized medium, we can see that the significant increase into the production was observed. That is a 8.92% of increase, which is the good for the industrial production also. Then we had, next please, we had purified this enzyme. Next please. Purified these enzymes and using the SDS page molecular characterization technique, we had found that the protease is having a 30 kilodalton weight, which is resembling with the previous literatures. Next, please. So these are the 4 different common phytopathogens. Again, these is the supernatant of the broth and the purified culture were used. Next, please. Yeah, here you can see the zone of inhibition due to the action of the protease onto the fungal phytopathogens, and it has inhibited the growth. Next, please. As well as these are the different plant growth promoting strain. In first side, you can see the zone, yellow zone, which is showing that siderophore production for the ion chelation by the ion chelation, then indolic IAA production and hydrogen cyanide production. Next, please. Yeah.
Professor, done. I think we should go to the question answer session now. We have
sure.
Too many questions to answer.
Sure.
For your presentation. Questions will be displayed on your screen. The first question from is Dheeraj Shekhawat. What is the yield of the proteins? Can you see the question on your screen?
Yeah. So the yield of protease I had shown into the table. Before to the optimization, it was something. It was 121 units per ml into the unoptimized. That is after the one variable at a time approach, and after the statistical optimization, it was 132 units per ml.
Okay, thank you. We have one more question.
Later. Yeah.
We have one more question from the same
please.
author. Why are isolate screened for cellulolytic activity?
Yeah, actually, the fungal cell wall is composed with the different polymers, and the cellulose is one of the content. So we were objecting to degrade all these components. So we had focused on the cellulose also, and later we had focused on the protease only.
Okay, we will take the last question now.
Please.
The question is from Deepika Singh. Can we use this method for the isolation of amylase enzyme?
Yeah, you can use, ma'am. But actually, we have to do some variation in media, which will be means I have another paper on that also for the amylase production. With the media is slightly varying because the amylase has to be produced. We have to just change the carbon source in which the amylase could be produced for the degradation by the enzyme, by the microbe. So you have to just change the carbon source.
Okay, fine. We will take the last 2 questions. The question from,
please. How significant is the protease yield compared to the reported values in the statistical? Sir, we had given this paper for it is in the communication, and you can see that these papers, these reportings what we had given is resembling with the maximum reports which were already published, but ours one is having the more yield as compared to the others, and we can say that the statistical approach is best as compared to the one variable at a time approach.
Okay, and the last question, please. It is visible on your screen.
Yeah, sure. Sir, you carried out like a combination. What is the difference? In placket Burman design, initially what the components we are selecting for the production, for that they will give you the 12 different sets of the experiments, in which we have to find the best combination of the variables, and later by using these readings we have to put that yield or the experimental results into the CCD, and in CCD they will give us whatever the variables were significant into the placket Burman. Of that significant, what is the concentration is significant. So in placket Burman we can find out only the combination with the zero value of the variables, but into the CCD we can find the actual value or what should we use for the maximum production. So actual concentration we can able to find with the help of CCD.
Thank you, sir. Thank you for your time. Thank you very much for the nice presentation. Thanks, sir.
Thank you very much to all of you for the patience.
So we have the next presenter on board with us, Chandrakala Sonawane. Ma'am, you can start your presentation as soon as your PPT is visible on the screen. Ma'am, there is some connectivity problem with your audio. Please verify it.
No, sir.
Okay, fine. Please proceed.
Yes, hello.
When the PPT is visible on your screen.
Sir. Okay. Hello. Hello.
Please go ahead.
Yes, sir. Good afternoon.
Please go ahead. Good afternoon.
Good afternoon, everyone. I am today presenting the development of functional food product from citrus fruit waste. Next slide, please. Next. Next slide. Next. What production of the lemon combined with the lime was 19.4 million tons. India first rank in the world for the production of lemon with 3.1 million tons in 2000. Lemon is an important medicinal plant of the family Rutaceae. One of the most important
your voice is breaking.
Citrus fruits. Hello. Hello.
Your voice is breaking.
Hello.
Go ahead, please.
Hello.
Please go ahead.
Citrus fruits are highly worldwide. Fresh produce. Most of the peel is discarded as a waste, but we utilize the new innovative that is the functional food. Most of the citrus peel contain antioxidants, that is the vitamin C, phytochemicals, and most of the dietary fibers also. Next, due to their low cost and easy availability, type of the waste capture offering the significance of low but nutritionally dietary supplements. Investigations have demonstrated that the citrus peel has an excellent source of vitamin C, phytochemicals, and also has the antimicrobial effect against the various food spoilage microorganisms. Thus, the present study and attempt was made to develop the functional food product from citrus fruits peel and their physicochemical property. Next slide, please. Next. 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 The material required: lemon peel, orange peel, salt, sugar. And then equipment required: that is the cabinet tray dryer, cutting, pressing machine, sheeting roller, refractometer, then juice extractor and big pot and this big spoon. That is the flow chart for the preparation of uh, citrus vadi. First we have to select the lemon and orange, then washing, then cutting, then removal of extracted juice, then after take the peel in a SS cooker and cook up to 20 minutes. Then we have to take the fine pulp. Another way, this fine pulp additional to cook to add the sugar and salt and cook the up thick consistency and getting the end point 50-degree breaks. And then after we have to cooling, then making the small ball, pressing, sheeting, then half drying at the temperature of 50 to 60 degrees Celsius for 1 hour, then give the perfect shape with cutter, then drying, final drying at the temperature 50 to 60 degrees Celsius for 12 to 15 hours, and then packing, and then after we have to store. Next slide, please. Next, next, next. Hello. Next, next slide, please. Next, next slide, please. Hello. This is the to development of lemon, getting the lemon peel, then peel, and we have to finally next, next slide. Then physicochemical we have to check on, that is the weight, peel percent, moisture content, fiber content, total soluble solid, titrable acidity, and organoleptic evolutions. The in which we have to calculate the weight. Take the 10 citrus fruits was measure and then calculate the average weight. Percent peel by subtracting the final weight of the peel from the weight of fruits. Then in which to calculate the moisture content. Then also we have to calculate the fiber content by the sample acid treatment AOC in 2024. Then result and conclusion in which we have to calculate the result, that is the protein 9.42%, fat is 4.9%, ash content is 6.2%, fiber is the 15.18%, peel percent is 40, and average weight is the 36%. Acidity is 0.15%, TSS is 15-degree Brix, and moisture content 7%. Next, in above table we have to conclude in the citrus come under the category of highly nutritious foods with the therapeutic value. The citrus fruits can't be consumed directly due to bitter taste, so processing is the best option to development of innovative product. Due to processing, it is a sweet, palatable, digestible, and easily consumable. Citrus is an excellent source of vitamin C, phytochemicals, and peel also acts as an antimicrobial effect against the various spoilage microbes. So, preparation of citrus vadi is the low cost technique, easy to handle, and require to no special storage condition. Small entrepreneur and the rural woman can easily prepare at the home, and citrus type of the fruit is available throughout the year. In this way we have to make the innovative functional food product from citrus citrus peel. Thank you, sir. you.
Some questions. Hello. Ma'am, you are not audible. There is probably.
Yes, sir. Hello. Hello, sir. Hello.
Yes, ma'am.
Hello.
Can you hear us?
Yes, sir.
So we have a question. The question is visible on your screen.
Which one, sir?
Can you please answer the question? Visible on your screen.
Yes, sir.
How does this lemon body compare with any ideal or a normal food?
Yes, sir. We have to compare the in market available so many type of fruit leather prepared from the citrus fruits, just like mango body. Just like we have to prepare same taste and nutritional point of same, just like mango body.
Okay, fine. The last question we will take. The question is, what properties do you study under organoleptic evaluation?
Yeah, yes, sir. We have to analyze the organoleptic evaluation, just like we have to color, flavor, taste, oral acceptability, color, 9-point hedonic cell sensory evolution, and in which we have to evaluate the oral acceptability of that food.
Thank you, ma'am. Thank you very much for the nice presentation. Thanks a lot.
Okay, thank you, sir.
The next presentation. Hello, good afternoon, Mr. Thorat. We will start with your presentation. Am I audible to you?
Yes, sir.
Okay, fine. I will display your PPT. As soon as it is visible on your screen, kindly proceed with the presentation.
Yes, sir.
Please proceed with your presentation.
Start करूँ, sir?
Yes.
Yes. Myself, Thorat Rameshwar. Today, my presentation, research and oral presentation topic on banana pseudostem in food application used as nutraceutical significance. Next slide. Contents. Next slide. Banana pseudostem introduction. India is the largest producer of banana next to the mango. Major producing states are the Maharashtra, Kerala, Tamil Nadu, Gujarat, Bihar, West Bengal, Assam, Karnataka, and Andhra Pradesh. Banana is the most important fruit crop. Banana plant biomass waste, that is pseudostem, contribute 30.81%. The annual production of banana in India is 26.2 metric ton. Banana pseudostem appears to be rich source of fiber, carbohydrates, and minerals. Banana pseudostem is also good source of potassium and vitamin B6 pyrotoxin. Health benefits of banana pseudostem. It is helps in flushing out of toxins from the body. It is very effective against kidney stone, It is very effective against pain relating to the kidney stone, helps in removing fat from the body, help in digestion, treats urinary tract infection. It is also very effective against anemia because it contains iron and vitamin B6 pyridoxine. Next slide. My research objective is preparation of banana seed stem powder by using two drying method. First is sun drying and tray drying. And second objective to study the nutritional composition of prepared banana seed stem powder. Next slide. Materials: banana seed stem, knife, tray dryer, packing material. Next one. Methodology: the preparation of banana seed stem powder. First is freshly collected mature banana seed stem and washed with potable water, and then next is peeling, and next one is a white colored pseudo stem cut into the small pieces. Next is next slide. Boil for 15 minutes, drying, sieve, and prepare banana seed stem powder. Next slide. Show the picture. First picture is raw banana seed stem, and second is peeled banana seed stem. Next one is white banana seed stem for eating, and next one is drying for the banana seed stem powder. Prepare the banana seed stem powder. Next one is analysis result of raw material, moisture content per hour, sun drying and tray drying. Show this. Next one. The higher drying rate constant was result from the higher amount of water removal with increasing drying temperature 14 to 70 and sun drying. Because of this higher moisture ratio is decreases, the mechanical drying of performance was better than the sun drying. In the sun drying, externally dried particle added. It is a long consuming process. Next one. The 60-degree temperature was better of other temperature. In this temperature, banana seed stem powder without any irregular burning. 60 degree centigrade is good in nutrient and color comparative to other temperature should show in below table. Increase of thickness, it was found that the thinner wear and slightly the faster drying rate. The 60-degree temperature, the color of banana seed stem powder very low brownish color and black dark spot as compared to other temperature. Next one. The nutritional value of banana seed stem powder. Banana seed stem powder, the parameter moisture, fat, protein, fiber, ash, carbohydrate, it's higher quantity, and major present in banana seed stem in minerals contains sodium, potassium, iron, calcium, manganese, phosphorus, magnesium. It is very important component in mineral banana seed stem powder. Next one. Conclusion. The taking the experimental result of the present investigation into the consideration, it can be concluded that the produced powder of banana seed stem, which normally considered a waste material in banana plantation, drying of dried banana seed stem could increase the shelf life of quickly perishable banana waste material, and it's suitable to incorporate in various recipes could be accepted to give health benefit in order to utilize the powder from in the number of value added food products such as bakery products, that is bread, biscuit, cookies, as well as in dairy products such as chicken, paneer, cheese, etc. To fortify these products with fiber, total carbohydrates and minerals, and the banana seed stem powder fortified with many food products and high fiber content, that is nutritional value of the product are higher.
Thank you, sir. We will proceed with the question answer session now.
Sir, एक minute. And the एक minute, sir. Banana pseudostem powder is one of the best method to preserve and improve the nutritional quality of the food product. And I one could think of new product development through the addition in the bakery, dairy product of banana, natural healthy product, which may also be served as appetizer. And banana pseudostem is waste material, and it is used food and has many medicinal properties, as well as high blood pressure control, dissolving the stone in kidney, weight loss,
Sir,
etc. And the sir.
Sir, thank you, sir. Thank you, sir, for your nice presentation. We will quickly take a few questions. The question is on your screen. Specific variety of banana is selected on what basis?
Yes, sir.
On what basis a specific variety of banana is selected?
No, specific.
Okay, then we will switch to the next question number 2. How to select a banana variety? On what basis do you select a banana variety?
Banana variety is Ardapur.
आप हिंदी में बोल सकते हो, आप हिंदी में बता सवाल का जवाब दे सकते हो.
Many varieties use, sir, banana pseudostem. That is, banana pseudostem एक ऐसा पार्ट है जो हम जो फेंक देते और उसको हम यूज भी नहीं करते, जो कि हम उसका यूज अच्छी तरह से उसका पाउडर बना के कर सकते, जो कि जो बनाना pseudostem रहता उसमें का जो व्हाइट पार्ट रहता वो पश्चिम भारत में जो अपने बटाटा जैसी सब्जी रहती उसमें वैसे खाने लायक बनाते हैं वो उसी से हम बनाना pseudostem का पाउडर बनाते, जो न्यूट्रासिटी के लिए, जो अपने डिसऑर्डर, हाई ब्लड प्रेशर, किडनी स्टोन, इस पे अच्छी तरह से.
Sir, thank you very much for your presentation. Thank you very much, sir. We are grateful to you for your time. Thank you.
Thank you, sir.
Good afternoon, ma'am.
Good afternoon.
This is Dr. Marine D'Souza. Ma'am, you kindly proceed for the presentation as soon as it is visible on your screen.
Yes.
My kind request to all the authors, kindly put the emphasis on your materials and results. Do not waste your time in explaining the introductory part. You are only given 6 minutes to explain your research work. Ma'am, you can proceed for your presentation.
The slide, okay. Good afternoon. My name is Dr. Myrene, and my topic for today is the anticariogenic activity of galls of Quercus infectoria against oral pathogens causing dental caries. So an insect gall is something which is formed in response to a kind of infection, invasion of the insect into any plant tissue, resulting in a neoplastic growth. And because of this, we have a large number of nutrients which are pulled into this gall, causing a kind of nutritional sink which is formed that enables the insect to grow. So obviously this gall will now be having a lot of nutrients which can be used for many purposes. So we see that it is ethnomedically very important, and in India it has been used to cure dysentery, gonorrhea, tonsillitis, hemorrhage, impetigo, and also to resolve postpartum uterine elasticity. So the Quercus infectoria gall has been known maybe in Ayurveda to Ayurvedic wisdom. We are trying to promote local and indigenous use of this product.
Okay, so coming to what exactly is the work about, we see that a number of orodental problems affect people, but though these are not very fatal, they do have aesthetic issues. And by using a number of conventional methods, which are not only expensive, they can also they have a lot of chemicals which would cause abrasion of the tooth surface and can cause sensitivity of gums as well as the tooth. Therefore, there's a need to use sustainable products as well as something that is accessible, low cost, and has been used in our tradition for many years. Next slide, please. Next slide. So a quick recap of the kind of work which was done and the results obtained. The extracts are prepared in 5 different solvent systems, differing in polarity, such as ethanol, methanol, acetone, ethyl acetate, and water. 10% extracts were made. It was centrifuged at 8,000 RPM for 15 minutes. And prior to storage, we sterilized it using a 0.22 micron porosity syringe, and then it was stored until further use. So the phytochemical screening of these gall extracts showed that the methanolic extract was rich in most of the bioactives, particularly something known as glycosides, which previous studies have reported to have a lot of orodental implications. So that is why the methanolic extract was used for further studies. Next slide. Next slide. So previous slides, sir. The clinical isolation of the organism was done from an infected tooth surface, a person suffering from dental caries. And after scraping of the plaque, it was taken into thioglycolic broth, streaked on brain heart infusion agar, and colonies were allowed to grow for 24 hours at 37 degrees centigrade. A number of colony characteristics, we got a number of isolates, actually around 20 isolates were seen. But and based on colony characteristics, we narrowed down colony 6 and continued with biochemical tests as well of these. So looking at the colony characteristics, it was seen that 75% of the colonies belong to Streptococcus genus, indicating that Streptococcus is actually the main player, especially as a primary colonizer. So during any plaque formation on the tooth surface, it starts off with Streptococcus, followed by the invasion of all other types of species. So if we are able to prevent colonization by streptococci, our chances of preventing dental plaque would increase. Next slide. So first we had to prove that the isolates that we chose were able to form the biofilm, because only when the biofilm is formed, only then the plaque would start being produced. So we did the primary screening of all the isolates, and we found that isolate number 6 was one of the best that formed the biofilm. So we used this for further analysis. Now, if you see in the test tube assay, we used 3 ml of BHI broth containing 1% of glucose. Overnight culture of isolate 6 was inoculated into it. It was incubated for 48 hours at 37 degrees centigrade, after which the media was decanted and PBS was used for rinsing, and was stained with 0.1% of crystal violet to see to check for the biofilm formation, which was seen in test tube B. After this, we performed growth kinetics. Planktonic growth analysis was done, in which 50 microliters of isolate 6 was thrown overnight and then inoculated into 5 ml of LB media, incubated for 37 degrees centigrade, and it was read every 30 minutes. It was seen that curve was obtained. Next slide, please. Next slide. Antimicrobial activity was done. Previous slide, sir. Antimicrobial, previous slide. Previous. Antimicrobial activity was performed by agarose gel diffusion method, and it was seen that when we compared this with the control chlorhexidine, we got good inhibition of the bacterial growth. Next slide. Next slide. MIC and MBC studies were also performed. It was seen that MIC, which is the lowest concentration of extract showing turbidity after 24 hours, the range was between 0.16 to 0.31 mg per ml, which was very good by means of study. And MBC, which is
the subculture of the organism was also correlating with that of the zone of inhibition. Next slide. Next slide, sir. So inhibition of, we did two methods of studying of inhibition. One was a qualitative assay using shell assays. So here we see that the control did not have ability to prevent the plaque formation, whereas the sample which was treated with the Quercus infectoria extract overnight for 10 days showed good inhibition. So this was done within with the media, with BHI media. So the nutrients, everything was available for the growth of the organism. But in the presence of QIE, we did not see coloration, indicating that the biofilm formation did not happen. Previous slide. This is a quantitative assay to prove biofilm formation inhibition, in which we use microtiter plates. For the microtiter plates, isolate 6 was grown in BHI containing 1% of glucose. Overnight culture was taken, 33 microliters of the broth with the QIE extract was added, and it incubated for 37 degrees and washed with 1%, washed and then stained with 1% of crystal violet. It was seen that with the increasing concentration of the extract, there was better inhibition or reduction percentage in the growth in the biofilm formation in our microtiter plates. That's all, sir.
So you are done with the presentation? Yes, we will go to the question and answer session.
So right now, this extract can be used for formulating mouthwashes and toothpaste. We are working on this. I'm working on this with a dental college.
Somebody has asked you a question. What type of controls you have used?
We use chlorhexidine, which is routinely present in mouthwashes to see zone of inhibition for the zone of inhibition studies.
Okay, we will take the next question, which is, you can see the screen.
Yes, sir. Is anti-cancer activity tested on or antibacterial activity? The title is Anticariogenic activity. Cariogenic would mean dental caries, dental cavities. It is not anti-cancer, it is anticariogenic.
Okay,
and
fine.
we use antibacterial studies.
Okay, and the last question.
Yes.
The reading of MBC.
MBC. So can you show the slide, please? Slideshow, sir. So from MBC studies, we saw that Staphylococcus was most susceptible to QIE extract as well as chlorhexidine.
Is this you want?
The MBC and yes, sir.
Can you see your presentation, ma'am?
No, sir, not yet. So MBC values were ranging from 0.16 to 0.131.
Can you see the presentation, ma'am?
Yes, sir. So, ma'am, Dheeraj ma'am, the lower table is the one for MBC.
So we'll go to the next question now.
Yes, sir.
Thank you, ma'am. Thank you very much for your
Thank you, sir.
presentation. Thanks a lot.
Thank you.
अभी हम इसमें से कौन प्रोफेसर को ले लूंगा मैं. I will take this professor. He has been waiting since morning. This.
Hello. Hello, am I audible?
Hello, ma'am. Palvi. Sri Krishna.
Yes, sir. Yes, sir.
Okay, ma'am. I will be sharing your presentation with you as soon as it is visible on your screen. Kindly proceed for the presentation.
Okay, sir. Okay, sir.
Please go ahead with your presentation.
Okay, sir. So, good afternoon, all of you. I'm here myself, Kamdepalli Sri Krishna. I'm working as a assistant professor in ACFT Bid Maharashtra. So we are. I'm going to discuss on topic development and recipe formulation of probiotic pomegranate drink used in digestion. Firstly, no, without going to the topic, first what is the role of pomegranate. What is the role of pomegranate probiotic drink? So why we are going to produce it? So why we are going to use this probiotic emerging technology? And next one is why we are using this pomegranate fruit. So firstly, what is probiotic? Probiotic is a use of microorganism life form. Probiotic is the concept of using microorganism in a life form for the purpose of digestion. So for the purpose of digestion. Why digestion? Why digestion is important? Many of us don't know what is the role of digestion improvement. Uh, Hello. So
Yes, yes, go
probiotic
ahead.
products in the market are mostly based on products such as yogurt, cheese, and fermented products. However, lately there has been an increased demand for non-dairy products due to various reasons such as allergens, lactose intolerance, high cholesterol content, and consumer turning to more natural products. So fruit juices are considered as an appropriate new substrate for probiotic delivery. The next concept is prebiotic. Probiotic are defined as live, single, or mixed microbial culture able to improve the composition of gut microbial flora to improve various microbial flora. So various probiotic strains have been categorized, mostly belonging to like Leuconostoc, Lactobacillus species, Pseudomonas, and other. Those are listed below. Next slide, please. After the probiotic, prebiotic concept is prebiotic is a food for probiotics. So we are using prebiotics for growth of probiotics. And the next concept is symbiotic. Symbiotic is a use of combination form of probiotic and prebiotics. So these are the three basic concepts. So, uh, so next come to the objective. Objective is of my topic is preparation of probiotic pomegranate drink that we are using it mostly for digestion, improvement digestion purpose. Sensory. Next thing is we evaluated this drink by sensory evaluation by use of 9-point hedonic scale. Next slide, please. So this is the methodology. First is selection of fresh fruit, that is a pomegranate. Pomegranate, why pomegranate? Pomegranate is which is a super fruit that we all, most of us that is don't know. So first point is selection of fresh fruit, then washing and cleaning. And next is preparation of juice. Store at 4 degrees Celsius before to use. Why we are going to store at 4 degrees Celsius? Because there are much more time needed for inoculation. So why it is last time for there is last time for inoculation, because in that first we are going to produce a microbial strain, that is a inoculum, that is isolated of Lactobacillus bulgaricus and Lactobacillus plantarum isolate, that is strains. So next to the strain, next point is inoculation in prepared freshly prepared before isolated the selected microflora in 100 ml juice. So after the inoculation, the next one is fermentation incubated at 37 degrees Celsius. After 4 weeks, beverage is ready to use. 4 weeks. 4 weeks. Firstly, the this 4 weeks is a large time period. So before to that, when we inoculated a microbial culture in prepared juice of 100 ml. So this is finally after that it is we got a fermented or final product in 7 hours, but the total procedure up to this. Hello. So after preparation of pomegranate probiotic drink, so it is stored at refrigerator temperature, and this is used for this is ready for use. So these are the composition of pomegranate juice, and next to composition of pomegranate juice, these are the application of pomegranate probiotic drinks. First application, why we are using probiotics in why we are using probiotic pomegranate drink is or we can say why we are using probiotics in food. So these are the applications. First is pomegranate is a antioxidant, anti-inflammatory, antibacterial, antiviral, and anti-tumor properties among others. So next one is why we are using. So next one is why we are using pomegranate juice. So it has a bioactive compounds such as phytochemicals like polyphenols and others. And the last one is fermentation of the juice with probiotic strains to provide beverages of high nutritional values and accepted digestive improvement. So next slide, please. So these are the results and discussion. The prepared probiotic pomegranate drinks were analyzed by 12 experienced people with a 9-point hedonic scale. We concluded that prepared probiotic pomegranate drink is a good and healthy for human consumption. So thank you. So topic is open for question and answer section.
Yes, thank you, madam. The topic is open for discussion. We can have questions. If anybody has any question. Madam, there is a question. How did you calculated colony forming unit or the inoculum for the preparation of this probiotic juice?
Hello.
Yes,
Hello?
Yes, you have used
Hello.
2 strains of Lactobacillus plantarum and Lactobacillus
Yes.
acidophilus.
Hello.
So I just want to know. Hello.
Sorry, ma'am. I'm not getting the question.
The question is, you have inoculated lactobacilli culture in the pomegranate juice. So how did
Yes.
you determine the total viable count? What was the quantity of inocula?
Hello. Sorry, ma'am, there is a range problem.
Okay, can you see this next question, which is displayed on your screen? What about the stability of the product?
Ma'am, if it's our stability of product, we can, yes, it has a good stability of product. If we keep in, or if we store it in good condition, or favorable condition, like refrigeration, so we can stable it, or we can store it for a long time.
Yes, just quickly give the answer for the next question. That
Yeah,
do
sure,
you
ma'am.
have idea about the viable cells content in the final preparation? Do you have any idea about the viable cells which will be there in the final preparation?
No, ma'am.
Okay, and the one more question is there any comparison with the milk-based probiotic? Have you
Yes, ma'am,
compared
we
your
can
study?
compare it. Yes, ma'am, we can compare it with milk-based probiotic bacteria. Uh, as compared to milk-based bacteria, as compared to milk-based probiotic, we can go for fruit-based probiotic. Why? Because mainly there are in many people there are lactose intolerance problem, so issues are like this. So that why we can that's why we should go for fruit-based probiotics.
Thank you so much
Yes,
for
ma'am.
your presentation. Now we'll have the next presentation.
Good afternoon, ma'am. Ma'am, could you please unmute yourself?
Good afternoon.
Yes. Kindly proceed for the presentation as and when the PPT is displayed on your phone.
Yes, yes.
Ma'am, please proceed for the presentation.
Yes.
Just a minute, there's some problem with your PPT. I will try to open again.
Okay. Okay. Hello.
Yes, yes. Please proceed for the presentation. The PPT is visible on your website. Please go ahead.
Hello. Good
Ma'am, please
afternoon,
go
one
ahead.
and all present here. Thank you, sir. Good afternoon, one and all present here. Myself, Prachiti Mohri, and I am going to present my PPT under the theme Natural Product and Drug Discovery. Next slide. Title of my presentation is Aloe Vera, a Natural Product as an Anti-Inflammatory Herbal Remedy. Nowadays, drug discovery using natural product has gained considerable attention, and natural products which are act as traditional medicine as well as therapeutic agent too are more useful. And aloe vera exhibit all these characteristics. In our study, we used aloe vera barbidinosis, one of the species of aloe vera which is most commonly and freely available. Thus, due to all these advantages, we aim to study aloe vera as an herbal drug for treatment of osteoarthritis. Next slide. Can you please skip that slide and go to next slide? Now, as we all see in this slide, this is the structure of anthraquinone. Now, what is anthraquinone? Anthraquinone is the phenolic compound mainly present in aloe vera, and anthraquinone constitutes of aloin and imodine. Now, aloin and imodine exhibit its anti-inflammatory activity due to their polyphenolic structure. Next slide. Now we will see how aloe vera shows anti-inflammatory activity. Now inflammation is the innate response of body against any injury, and aloe vera exhibit anti-inflammatory action by this extracellular signal related pathway. In this pathway, aloe vera gel extracts inhibit cyclooxygenase pathway by producing arachidonic acid. Simultaneously, it reduces the production of COX-2 prostaglandin, and thus it suppresses the level of TNF factor like NKP and JNK, and thus shows anti-inflammatory activity. Literature review and literature studies reveal that aloe vera contains novel anti-inflammatory compound named as C-glycosylchromone. Next slide. Now, question arises, what is osteoarthritis? Osteoarthritis is caused by mechanical wear and tear in joints, and it shows mild inflammation, and it is solely limited to joint. There are several forms of arthritis, but most common form is osteoarthritis. Next slide. These are the symptoms of osteoarthritis like pain, stiffness, redness, swelling, loss of movement of joint. Next slide. Next slide, please. These are the causes of osteoarthritis like obesity, excess of junk food, vitamin D deficiency. Next slide. Now, as we all see, we can see in this graph, it is evident that as the prevalence factor increases with the age, and women are more vulnerable to OA as compared to men of same group. Next slide. So finally, I would like to conclude that associated with more side effects, and they are expensive as compared to natural drug. While natural drugs are friendly, thus there is a need for an alternative treatment with safer compound, and thus the drug and could be preferred for the choice of osteoarthritis. Thank you.
Thank you, ma'am. We will take some quick questions if we're not having any questions. Thank you, ma'am, for your nice presentation. Thank you very much.
Thank you.
Hello, Professor Chetali, can you hear me? Can you please unmute yourself? Ma'am, can you hear me? You are not audible. Professor Chitali Amre. We cannot hear you in the studio. Okay, we will proceed to the next presentation. Hello, Professor. Good afternoon. Can you hear us?
Yeah, good afternoon. Can you hear me?
Yes. We will be proceeding for the presentation. You kindly proceed with the presentation as soon as the PPT is visible on your display.
Sure.
Ma'am, please complete your presentation in 5 minutes.
Sure. No problem.
Go ahead, please.
Good afternoon to one and all. Thank you to the organizers for this international conference for giving me this opportunity. My topic is screening of successive extract of Cassia abscess seeds for antibiotic potential. I am Professor Ekta from JCRC University, Department of Biotechnology, Jaipur. Next. This is the abstract. Next. Yeah. Introduction. If you all see that traditional medicines have been known for many centuries for the part of or use of another kind of ailments. Next. If you see these kind of plants out of which I have focused on the Acacia abscessus, if you will see, it's a ethnomedicinal use plant having the traditional properties against the bronchitis, asthma. Its seeds are very much useful and commonly known as the chaksu seeds. Next. What the material methods we have used the extraction procedures from successive extracts where the Cassia abscess seeds were shadily dried and then crushed, powdered, will use for the successive extraction. Next. This is the distillation unit rotavap where you can see the evaporations were further performed so that the bit of the solvent will not be there in the samples. For the next. First, we collect the sample and the preparation of the plant sample in the successive extraction, which will be further performed with the antimicrobial efficacy and the antioxidant activity. Antimicrobial on the basis of this diffusion method of the golden Bavaski and antioxidant of the Tikayo 1994. Next. These are the few test microorganisms which are used for the bioefficacy. Next. This is the process which is the normal where we first prepare the culture media. These media were autoclaved and then put in the 37-degree temperature in the incubator, and further they were used for the various kind of performance of the disk diffusion method. Next, this diffusion method is basically performed with the extracts with the 5 mg and 10 mg per disk, and these discs were be incubated, or we can say put on the slides where the already inoculum was prepared and put, and this incubation was done for 24 hours, and after 24 hours the activity index or the inhibition zones were seen. The inhibition zones were seen with the standard drug, that is a tetracycline, and this activity index was calculated. Next. Antioxidant assay was also performed by the this diffusion method. Tikayo et al. Method. In this, radial scavenging activity was performed where the standard was ascorbic acid was used and the percent inhibition was calculated. Next. The results. Next. We can see this is just the outlay about the what radial scavenging activity performed and how it is useful. Next. Result and discussion. If you will see the clear cut while performing against the various microorganisms, S. aureus showed the promising result as compared to the others. So Cassia abscess seeds are giving a very good result against the S. aureus. All the extract have showing the good inhibition zones which are present in the form of the peaks. You can see in the pink zone or the pink graph, where we can see one or the another type of inhibition is present, and this can be used nowadays also in the COVID-19 situation where the respiratory problems are there. That COVID is also belong to the respiratory tract infection, and S or S is also inhibiting. So that means cassia abscess also can use for the same. Next, if we see the next antioxidant result, if we see, they are not the methanolic hot extract of C. abscess show antioxidant properties, but that is not as much as the ascorbic acid standard possess. But closely observations will also show that even the cassia abscess seeds at a very low concentration is showing the promising results. That is the inhibition rate to the 87.5%. It is commonly known as the Chaksu seeds. So we can use this further as a part of a diet also. Next. This is the graph where comparison is there. So discussion in last we can see. Next. Discussion we can previous one. In this, we can clearly say that C. abscess seeds show not much antimicrobial efficacy, but as per we can see some of the extracts show promising activity, like in K. pneumonia, inhibition zones were very good, and S. aureus, against S. aureus it is showing. Thank you very much.
Thank you, ma'am. We will go to the question answer section now. I'll take some quick questions.
Sure.
Here is the question on your screen. Which ethnic community using the seeds as herbal medicine in India?
Here in tribals, Mongia district is using. If you can see, there is a Mount Abu region. There is the Mount Abu Saharia place. There people are residing. We interact with these kind of people. If you go to our few of the publications, you can clearly see that we have outlated that what kind of tribes which are using these kind of seeds. So they are doing that. Sure.
Okay, thank you. We will take one more question,
sure,
if there is anyone. Thank you, ma'am, for the nice presentation. Thank you very much.
Thank you. Thank you.
Good afternoon, ma'am. Could you please unmute yourself? Ma'am, you are not audible. Could you please check your microphone?
Am I audible now? Am I audible now?
Hello. Yes, you are audible, but there's a lot of noise from the background.
Yes, now is it fine?
Hello.
Yes, sir.
Ma'am, yes, okay, fine. Please proceed with the presentation as and when the PPT is displayed on your display. Okay?
Okay, sir. Yes, sir.
Ma'am, you have 5 minutes to complete your presentation. Please go ahead.
Good afternoon, everyone. This research was conducted for the evaluation of anti-cataractogenic activity of Spinacia oleracea on glucose-induced cataract in isolated goat lenses. Next. Cataract
all know, it's just the loss in the transparency of the lens, and an opacity develops due to the loss of the proper arrangement or the structure of the crystalline protein. Next. So in order to meet that particular aim, our experiment was divided firstly into having a methanolic extract of spinach leaves, and then followed by a photographic evaluation of all the 5 sets that we had created. This cataract, that it has numerous mechanisms. First being the lens swelling, and that can be due to an imbalance in the pump system, namely sodium potassium ATPase pump, then sugar cataract, which we are going to deal in detail. And the other risk factors include diuretic, cigarettes, alcohols, and steroids. Next. Diabetic cataract, it's a major secondary complication of diabetes mellitus, and polyol pathway is known to play a major role in this. So as we can see in the figure, that glucose gets converted to sorbitol, and sorbitol in turn gets converted to fructose in a normal healthy lens. But in case there is a cataract development, sorbitol tends to get accumulated there, leading to osmotic stress. This leads to electrolyte imbalance and membrane damage too. Next. Next. We can go to the result one, yes. So this picture clearly depicts our experiment where we had initially divided the lenses into 5 sets. First being the control one, which is a positive control, that is the 55 millimolar glucose is bound to produce a cataract. The last one, if we see, that was incubated with enalapril, a cataract preventing or controlling medicine. And test one had given a really good result when compared to the medicine that we had, medicine sample that we had induced. So carrying forward with group 2, we that is 100 μg per ml of spinach extract, we had incubated more of the lenses at 72 hours and carried forward the following assays. Next. In this major, let's start with the total protein content. Whenever there is in a cataract developed lens, there's bound to be less of protein content because of the sodium potassium ATPase imbalance, leading to a defective membrane permeability, causes a leakage of the protein. And spinach did a good job in increasing the amount. MDA, that is malin dialdehyde, and copper induce lipoprotein diene formation. They go hand in hand. And MDA, it's a stable lipid peroxidation product, and it was greatly reduced in a spinach extract. And lipoprotein diene formation, it's directly related to the absorbance. So, and when we observed the lens of that was incubated in spinach extract, a reduced observation of 0.29 was observed. GSH, that is reduced glutathione and glutathione reductase, are relatable. Glutathione, it is a major antioxidant present, and GSSG, its oxidized product, is converted to GSH with the help of glutathione reductase. So an increased amount of glutathione helps in the protection of the lens, and that's what we had observed with spinach extract. And peroxidase and hydrogen peroxide results are again related. Hydrogen peroxide is converted to water and oxygen in presence of peroxidase. So a lower amount of peroxide, that is hydrogen peroxide, is a good indication of our samples working well. Next. So in conclusion, next. In conclusion, we can say that our spinach extract was having a good anti-cataract activity, and it showed a dose-dependent effect. Thank you.
Thank you, ma'am. We will take some quick questions.
Yes, sir.
Any questions? Thank you, ma'am. We do not have any
Thank
questions.
you,
Thank
sir,
you for the
for
nice presentation. Thank you very much.
Thank you, sir.
We have Rutuja Sonali on board with us. Rutuja, could you please unmute yourself?
Yes, yes, sir.
Fine. So you kindly proceed with your presentation as and when you see the PPT on your mobile display, okay?
Okay. कंशु कंशु बंद कर.
Please proceed with the presentation. You have 5 minutes complete.
Sure, sir. Okay. Good afternoon, all. The topic for presentation is the isolation and a growth characterization of a halophilic fungi from a natural source. Next slide, sir. Okay, so a very small introduction. As I'm only interested in the halophilic fungi, so basically they are extremophiles, and the halophilic fungi are the one which requires a salt concentration for their growth. So as per the requirement of the salt, they are divided into a few types like a slight halophile, a moderate halophile, and the extreme halophile. Next slide, sir. Next. Next. Okay, so I will start with the objective. Now, as I said in a title that I am isolating the halophilic fungi, so the first part is the collection of the natural samples for the isolation of the fungi. A second part, once I get a sample, I did a isolation and a purification of the fungi for the further studies. And the third objective was to identify whether it is a halophilic or not. And after that, I was supposed to study the growth optimization of the isolated fungi. Next slide, sir. Okay, so these were the methods which I used. First, I collected the sample. So I selected two sample, a seawater sample and the air sample. Next, once I used the sample, I stripped the sample on a specific media, that is a potato dextrose agar. I incubated that for 3 to 4 days at a room temperature with a pH 5.6. Some plates were with NaCl and some plates were without NaCl. Okay, once I got some growth on the plates, I removed the fungus from that for a purification purpose, because I wanted to isolate each and every fungus separately for my further studies. Once I done with the isolation part, I did a macroscopic examination by using a staining method which is commonly used, that is a lactophenol cotton blue staining. After that, I did a growth optimization because I wanted to confirm that whether this fungus is a halophilic or not. And if it is halophilic, then among which concentration it sets, whether it is a whether it is extreme or whether it is a moderate. Okay, so for that I used an NaCl concentration which is ranging from 0% to a 25% with the same pH and a room temperature. Next slide, sir. Okay, so this was the result. So as I said, first I isolated the fungi from the sample. So I got 8 isolates. This you can see on a plates also. By looking at the growth morphology, you can understand that there are fungi. So I just separate out each and every fungus. Next slide, sir. Next, I did a microscopic examination. Next slide. Okay, so this is the last growth optimization. Now in this you can see I have named isolates depending upon the source, like H1 that is for the seawater sample and AR for the air sample. Okay, and I use the NaCl concentration which was ranging from a 0% up to the 25% with 2.5 intervals. Okay, and as you can see, up to 20% I have got a growth of the fungus. So so as you can see, at 20% there were positive sign I have given. The more positive, the more growth I got. Okay, so next slide, sir. Okay, so from this whole study, my conclusion is that as it is just a preliminary part, a preliminary research of my whole PhD, which I'm going to continue ahead. Okay, so first I isolated halophilic fungi from a seawater and the air sample. After that, I found whether they are halophilic or not. So for that, I did a growth optimization and I used a concentration of 0 to 25%, and among that, I found the fungus was growing from 0% up to the 20%. I did a microscopic examination, but I haven't identified that at a species level yet. I need to do it. And my further aspect is to isolate
Thank
or
you.
to get a bioactive
Thank you, Ms.
compound.
Rutuja. Thank you, Ms. Rutuja. You are out of the time. We will quickly take few questions. Thank you very much.
Okay, okay. Yeah, yeah, sure.
Madhuri Deshmukh is there. She's saying, I have a question, but she has not asked the question yet. Now, we have a question. Ratuja, can you hear us? What is the importance of isolation characterization of halophiles?
Importance.
What is the
Okay.
importance of
Sir,
isolation
actually,
characterization?
sir, actually, my whole research is based on. At the end, I'm interested in extracting the bioactive compounds from those halophilic fungus. And as per the various literature survey which I have read, I found that uh, this particular halophilic fungus is producing some sort of the bioactive compounds. But my main motto is to use these bioactive compounds on the multi-drug resistant microorganisms, which I am going to isolate in further studies. So that's why I selected specifically the halophilic fungi.
Okay, thank you very much. Thank you a lot. Thanks a lot for your time and presentation. Thank you.
Thank you, sir.
Hello, Miss Professor Sharda Patika. You are not audible. Could you please unmute yourself? Your microphone is mute, I suppose.
Yes.
Okay, fine. Thank you very much for your time. Kindly proceed with the presentation as and when the PPT gets displayed on your mobile phone. Number क्या 21. Number 21. ये है ना शारदा पाटेकर? Please go ahead.
Yes. Good afternoon, all. Myself, Patekar Sharda. I am from Department of Microbiology from KSK College of Food Technology, Bid. So today's my topic is the wheatgrass juice and powder as a natural and holistic plant medicine. So many drugs are commonly today are using as a herbal origin. So some are from used from the plant extract. So wheatgrass is nothing but a nature's finest plant medicine. So generally the wheat germinated over a period of 6 to 10 days. Generally called as the wheatgrass. And during this germination process, different types of vitamins, minerals,
and the phytochemicals, including the flavonoids, are synthesized into the wheat sprouts. So it will reaches the maximum antioxidant potential. So my objective of this study is to explore the presence of phytochemicals and the presence of flavonoids into the wheatgrass in order to the role of wheatgrass in plant medicine, as well as the proximate nutrient content into the wheatgrass, including the antioxidant activity. So the phytochemical analysis in the plants is a very important commercially due to the great interest for the production of different new drugs and for curing of the different diseases. So the process for the preparation of wheatgrass powder and the juice is generally first of all the harvesting of fresh wheatgrass is carried out. Then these wheatgrass leaves are removed for are washing for the removing of the microorganisms and soil, and then they are going for the sorting and cutting.
Ma'am, please
Then
explain your
next is
result section. We are running short of the time. Please explain your result and discussion section.
okay, sir. So the basically the study is carried out for the phytochemical analysis and for the proximate nutrient content into the wheatgrass juice and powder. So my result and discussion for the proximate composition, the moisture content into the wheatgrass that shows that 16.66% of moisture, and then it was found that ash content that is 7.9, fat percent into the wheatgrass powder that is 0.003, then protein content 27.267%, and the crude fiber content that was found 20.60%. So this was based on the different research studies and on the proximate analysis of wheatgrass. Then
Thank you.
different phytochemical analysis was carried out for the flavonoids, alkaloids, and the phenol content, as well as the antioxidant activity. So it is antioxidant activity is more important in this wheatgrass. The flavonoid found that will be a 161.10 milligram. As per the study, it was found 160.20 gram. So next is the antioxidant activity. So that will be a 73.93% for the that antioxidant activity will leads to the different curing of the anti-cancer agent, then anti-inflammatory. So it is beneficial. And then conclusion. So from the results, it should be concluded that wheatgrass should be consumed regularly as per the body's requirement, and different presence of the phytochemicals into the flavonoids, alkaloids, and phenol content will increases its interest into the plant-derived herbal medicines. So the richest antioxidant potential of wheatgrass also proves that the beneficial for certain conditions or certain diseases like anemia, diabetes, anti-cancer activity, anti-ulcer activity, or anti-inflammatory. So it is better to be add to wheatgrass onto the daily basis in our diet. So
Thank you,
on
ma'am.
the
Thank you.
rational
We will take some
evidences.
quick questions, madam. We are out of the time. Thank you very much.
Okay.
So
Okay,
the
sir.
first question is, could you please tell us about the qualitative phytochemical analysis which you have done so far? There, you have not mentioned in your post the kind of phytochemicals you have tested qualitatively.
Yes, sir. I have analyzed the phytochemicals, for example, the flavonoids, then phenol content. So that was found into the wheatgrass powder as flavonoids 161.10 milligram, then phenol content 0.50 milligram, and the antioxidant activity.
Okay. Thank you, ma'am. Thank you very much.
Thank you, sir.
Professor Someshwa? Professor Someshwa, can you hear us? Professor Someshwar Khandekar. Hear me?
Hello.
Can you hear me?
Yes, sir.
Okay, fine. I hope you have a good, proper connectivity.
Sir.
Is there a connectivity problem?
No, sir. Abhi.
Okay, fine. We will go with the presentation. We will go your presentation. Please proceed for the presentation when the PPT is visible on your phone.
Okay.
Please proceed for the presentation. You have only 4 minutes to present your PPT.
Okay, sir. Good afternoon, everyone. Today's I am discussing topic use of the amla in order to cure the arise major diseases. Please next slide. Next. Firstly, introduction. The avla is a very useful component fruit in India. The use of avla in different medicine, like of the this different medicine and medical good medical property. This avla is one of the fruits as before processing and after processing to maintain the nutritional value. This avla is a major production in Rajasthan, Madhya Pradesh, Uttar Pradesh, Maharashtra, and Tamil Nadu. This production of the avla in winter season, this avla production, this each and every season to available of the avla product, and this avla to maintain the good medicinal value, like the maintain the sugar level as a source of vitamin C, and to improve the medicinal improve the immunity system, and those level sugar, the major source of vitamin C, and to improve immunity system and to a good digestion on and to control the acidity. The natural source of the anti-aging present in amla. The Sanskrit name of amla is an amalaki. The use of different form of avla, like powder form, juice, paste, and to prepare the different type product and to increasing shelf life of this avla, like the candy, muramba, and the supari, avla supari, can be help to prevent the free radicals. The avla extract, the Japanese university is Japan. The scientists at the Faculty of the Pharmaceutical Science in Nagasaki University to discover the primary research as amla fruit extract to prevent the cancer cell and to free from the cancer, and to use of the avla to cure the different disease like scurvy, and free of the other disease.
Mr. Khandekar, you are requested to conclude your presentation. Kindly conclude your presentation. We will take some quick questions. So we don't have any questions from the audience. Thank you very much for your nice presentation. Thanks a lot. Thank you, sir.
Thank you, sir.
Ms. Chetali, can you hear me?
Yes, sir. I'm hearing you.
Okay, fine. Kindly proceed for the presentation as and when the PPT is displayed on your phone.
Yes, sir.
Please proceed. You have only 3 minutes to present your poster. Please go ahead.
Yes, sir. Good morning. Myself, Chaitali. My topic is about the drug discovery from the natural product sources. In the past decades, we can see in the past decades, the natural products in the pharmaceutical industries has declined owing to the issues of the lack of compatibility of the traditional natural products and through the high throughput screening techniques. And in this era, we can make them better, so we have to take some we I have decided to give some about the introduction and some conclusions about that. Take years to a decade for discovering a new drug very costly. We have some 7 steps that we can discover a new drug through screening techniques and very effectively. First is choosing a disease, choosing drug target, identifying biosafe and lead compound, isolation and purification, structural determination, and the herbal medicine. The first question arises in all our mind is that what is the drug development? I mean, how it occurs that the process of bringing a drug development in the market has been taken a lot of problems. But if we find the lead compound and some researchers find the lead compound, the process of drug discovery made very easy. The next is the natural products. If we see the natural products, richest source of the biologically active compound. Today's medicine obtained directly either from the lead compound and originally from the plant compounds. In short, a natural product is the chemical compound or a substance produced by a living organism that found in nature. Almost the 50% prescription given by the doctor, almost the 50% prescription medicine given by the doctor was also extracted from the plant compounds. First, choosing a disease. If we're choosing a disease, then we have to make sure that they does not affect the lowest economic countries like in the third world centuries and the first world centuries.
Ma'am, could you please
And I take
conclude your PPT? Kindly conclude
uh,
your presentation. We are running short of the time.
uh, according to the conclusion, if I say that the natural products are the derivatives of the different things, and the one of them is the plant source, and that is the morphite.
Okay, thank you. We will take quick questions from the audience. Thank you, ma'am. We do not have any questions from the audience. Thank you very much for your presentation.
Thank you.
Thanks a lot.
Thank you.
So, the last but one presentation, Rupali Mahadev Bathe. Ma'am, if you could hear us, kindly join the studio for the presentation. Okay, we will switch over to the next presenter. It is Dhruv Pandya. Hello, sir. Good afternoon. Can you hear me?
Yes, yes. Can you hear me?
Yes. You are very much audible. Kindly proceed for the presentation as and when the PPT is displayed on your phone.
Sure, sir. Sure.
Sir, you have 4 minutes to present your PPT. Thank you very much. Please proceed.
Okay. Sure. Namaskar, one and all present here. First of all, I would like to thank the organizers and speakers of the day who provided me this opportunity to deliver this content to the audience. Whenever we are talking about environment, at that time, pollutants are the major parameters that we have to control. And my research is on that remediation portion. Remediation means to treat, and phyto means the plant. Phytoremediation means we are taking that particular plants as media, and we are trying to remove the pollutants from the environment. So next, please. Okay. So next, please. Next. So main aim of the study is to remove the pollutants, which are the major pollutants like heavy metals from the soil, and we try to convert that infertile soil in fertile soil. And we have selected here Zinnia elangus as plant because this plant is annual plant and not a non-edible plants for every animal. Next, please.
In the methodology, we are trying to produce two different ways that plantlets of Xenia in vitro condition as well as in vivo condition. In vitro condition, we are trying to produce it in through micropropagation in plant tissue culture, and in vivo we are trying to directly producing within the soil. Next slide, please. So here the slide shows that in vitro and in vivo production of plantlets. After the production of this in vitro and in vivo plantlets, we are trying to potted it and treated it with heavy metal, and the metal was selected was lead, and we selected that concentration from 200 to 800 mg per kg in the soil. Next slide, please. After the 60 days treatment of lead, we found these results that in vitro plantlets having more capacity to uptake or accumulate or extract that particular heavy metal from the soil as compared to in vivo produced plantlets. Next, please. Even we also observed that growth parameter difference as the metal concentration increases, the shoot length, root length, as well as the number of flowers are decreasing. Next, please. Even yes, we also found that this soil analysis before treatment and after treatment. You can also see the difference before treatment and after treatment. That after treatment, the heavy metal amount is decreasing in the soil. Next, please. So these are the conclusions. So this is the key technique to remove the heavy metal from the soil. And in vitro produced plantlets, if we are using, then they are more capable to remove that particular pollutant from the soil. And one major thing after this research that we found, that generally the researcher after the research, they are burning that particular product and they are throwing in the dustbin, which is actually not proper way. Here we are trying to utilize that secondary product, the plant that we are having with heavy metal, that we are burning, and after that we are extracting that particular char, that is biochar, and that can be utilized for this tire industry and tube industry, so that secondary products can be also utilized after the research. So thank you very much. Any questions from
Thank you very much. I will get back to you once I get the questions from the audience.
Please.
Thank you, sir. We don't have any questions from the presentation. Thank you very much. Thanks a lot.
Namaskar. Namaskar.
So today, before I conclude today's session, I would like to place on record a big thank you to our speakers, Professor Liv from USA and Professor Ramachandran from Dubai. And last but not the least, our presenters, probably 20 odd presenters presented their research work in the form of PPT and posters. Thank you all. And tomorrow, we hope to see you again at 9 a.m. Till then, take care and bye.